Bom, muito bom dia a todos aqui e a todas aqui presentes, aos amigos que nos acompanham pela internet. Nós vamos conduzir esse evento na língua inglesa. So, good morning, welcome to the Genome 20+2 workshop. To open this session, I'd like to invite the president of FAPESP, Professor Marco Antônio Zago. Scientific Director of FAPESP, Professor Luiz Eugênio de Mello. And from the Fundo, Pitanga, Professor Fernando Reinach, please. Okay, so Professor Zago, you have the floor.
Okay. Good morning, my friends, everyone. This is a moment to commemorate a big success of Brazilian science, a success that was born in FAPESP. I wish to welcome all of you who came to this conference, students, researchers, speakers, and also people connected to the internet. And also, I wish to thank all who worked to organize this meeting. I will mention Ana Maria Camargo, Marianne Fons Luis, and Claudia Izik as representatives of a large group of scientists and FAPESP staff who worked in this meeting. This is one of the last events to commemorate the 60 years of FAPESP. Well, on the 30th of July 2000, the cover of the prestigious scientific journal, Nature, stamped the picture referring to the article with the complete sequence of the Xylella fastidiosa, a plant bacterium that was causing big losses to the orange plantations in the state of São Paulo, then went to infect vineyards in California, and today infects olive trees in Puglia, in Italy. It was the first phytobacterium to be completely sequenced in a period of less than 2 years, an extraordinary feat at that time. What is more relevant, this result was not obtained by one highly efficient laboratory and organized laboratory, but by a network of 35 laboratories and more than 130 researchers, most of them who had never sequenced even small segments of DNA. In addition to FAPESP, this initial push into genomics research in São Paulo had two partners: Fundecitrus, a research and education association created by citrus industry and farmers, and the Ludwig Institute for Cancer Research, a long-standing partnership that extended far beyond financial support. We will dedicate a special opportunity tomorrow to commemorate the partnership between FAPESP and Ludwig. Okay, 10 years afterwards, in July 2010, an editorial of Nature summarized the adventure this way. In May 1997, a pair of Brazilian scientists spent a weekend in the countryside discussing a bold idea. José Fernando Pérez, the science director at the São Paulo Research Foundation, FAPESP, had been looking for game-changing research initiatives. Biologist Fernando Reinach, one of his advisors, had a sufficiently adventurous plan: kickstart biotechnology research throughout Brazil by sequencing a genome. The duo pushed ahead to build the capacity for genomics and bioinformatics that Brazil was lacking, quickly organizing a team to conduct the project and then setting on the bacterium to sequence. FAPESP invested the equivalent of $12 million, largely dedicated to sequencers, computers, and reagents, while the team brought together and trained the researchers from a large range of fields to develop a broad and long-lasting set of skills and knowledges. Brazilian biotechnology has matured to the point at which the scientists are now players in the international stage. Perhaps more than anything, Xylella demonstrates the benefits of aiming high, because, and this I add and stress, because if you invest only in low-risk regular projects, the best results you pick up will probably be a correct regular paper. But if you aim high, you have the chance to really change the panorama. Thus, the program started in 1998 with well-defined and simple targets. It soon became evident that the same approach would be applied to many other living beings and situations. Even before the Xylella paper had been published, many groups organized to investigate animal, plant, and infectious diseases agents' genomes. Many of these people are here today. Some were invited to speak to us. More relevant, some of their students or the students of their students are here with us today. The skills and knowledge that we started to acquire at that moment are highly valuable today, as they have been, for instance, during the COVID pandemics. The legacy of that period extends to all fields of research and life sciences today. The next one, and the next also. Again and again. And the second or third generation of researchers of this lineage are now manipulating the genome to help treat diseases, as they are manipulating the genome to improve food production. As in this case, next one, of manipulating the cell of the patient to treat his own cancer. Thus, I wish to close stressing again the statement in Nature that Xylella demonstrated that the benefits of aiming high in science are worth. And finally, I wish to compliment all those who organized and those who were part of that special moment in the life of FAPESP and of science in the state of São Paulo. Thank you.
Thank you, Professor Zago. We will now hear the Scientific Director of FAPESP, Professor Luiz Mello.
Good morning to you all. It's a great pleasure being here today at this memorable event celebrating the 22nd birthday of anniversary of a major event in Brazilian science, which was the Genome Project led by FAPESP, as already mentioned by Professor Zago. I understand that that program has a mythical nature, and to speak about it, I will then switch to Portuguese, and in a manner that I might be more able to express and convey the ideas I have on this. Grandes feitos e grandes conquistas, como essas que estamos hoje celebrando, têm uma natureza lendária. Elementos centrais em uma lenda são os personagens, vários deles aqui presentes hoje. É do empenho e das histórias de superação desses personagens, cientistas, dirigentes da FAPESP, seu quadro técnico, que decorrem as conquistas que hoje celebramos. Central em uma lenda é também a trama, seus desdobramentos, suas intrigas, as sequências de sucessos e de fracassos. Nesse caso, finalmente coroadas pelas grandes conquistas e impactos duradouros. No projeto Genoma da FAPESP, todos esses elementos estiveram presentes. O programa teve forte oposição, havendo, por exemplo, os que defendiam que muito mais importante que gastar milhões desvendando a sequência genômica humana seria investigar a função dos genes no organismo. O tempo, em ciência, frequentemente não é imediato. O conhecimento se constrói e os frutos podem demorar, muito como os 60 anos de construção da FAPESP. Nesse programa, a FAPESP apostou em uma visão de longo prazo e visou não apenas sequenciar uma bactéria particular, mas também, e talvez principalmente, criar uma infraestrutura material e humana complexa e potente na área da genômica, cuja fertilidade foi evidenciada nas décadas seguintes na realização de inúmeros projetos de pesquisa básica e aplicada, conforme mostrados aqui pelo professor Zago. É muito fácil ser engenheiro de obra pronta. Nessa linha, na celebração de hoje, além de reconhecermos o mérito do programa brilhantemente desenvolvido por vários dos que estão aqui presentes, temos também que parabenizá-los pela sua tenacidade no enfrentamento das adversidades. A FAPESP é constituída por diferentes esferas de ação, com funções distintas e complementares, todas importantes e com mérito pelo seu sucesso. Mas é a força da ação da nossa comunidade científica, não apenas na execução das pesquisas, mas também enquanto formalmente representada na FAPESP por sua assessoria científica, na gestação dos programas de fomento que as viabilizam, que nos permite e nos permitiu dar saltos como os que celebramos hoje. Parabéns aos cientistas que fizeram o projeto acontecer. Parabéns às lideranças que não se dobraram perante visões de mundo imediatistas, fundamentadas em concepções equivocadas do andamento do tempo da ciência e da dinâmica imprevisível das interações entre pesquisa básica e pesquisa aplicada. Obrigado. Tenhamos um ótimo evento.
Thank you, Professor Mello. We now give the floor to Professor Fernando Reinach. Please.
Bom dia a todo mundo. Marianne me aprontou essa, não sabia que eu tinha que falar. Eu achei que eu ia só fazer número aqui. Mas, bom, eu acho que eu estava pensando duas coisas que eu acho importantes. Uma é essa coisa de realmente ousar. Eu acho que é uma coisa que falta muito na ciência brasileira, de realmente correr o risco e fazer projetos que parecem muito difíceis. Eu acho que eu tenho um sentimento, eu estou muito distante aqui, mas eu tenho um sentimento, vendo a distância, que esse ânimo arrefeceu um pouco. Eu não vejo, pelo menos eu não estou sabendo, de grandes projetos ousados, tentando fazer coisas realmente difíceis ou challenging, né? Eu acho que isso é uma experiência que a gente teve, foi fantástico, foi um dos melhores períodos da minha vida. A gente se divertiu muito durante 2, 3 anos. Passamos medo, passamos prazer, etc. Mas foi muito legal. E foi uma coisa totalmente conjunta, né? A gente nem pode dizer... A gente tinha o Simpson, que era o nosso líder, mas era um negócio completamente multirresponsabilidades, todo mundo ajudando. Não foi um projeto muito de cima para baixo, com um coordenador com muita autoridade, nada desse tipo. O que unia todo mundo era essa ideia de conseguir sequenciar o genoma. E a outra ideia era treinar as pessoas, né? Eu acho que o projeto ainda... Essa é a segunda coisa que eu queria dizer. Eu acho que o projeto não terminou, no sentido que eu não vi ainda, na comunidade acadêmica, um estudo sobre o impacto do projeto. A gente tem muitas coisas, como o Zago mostrou aqui, um número grande de papers, de outros projetos, etc. Mas a gente tinha, não sei quantos autores tinha lá, acho que tinha uns 100 e tantos autores no paper, né? A maioria de pessoas jovens. Eu não sei como quantificar isso na história da ciência, mas a gente devia fazer um esforço de quantificar, porque um dos objetivos principais era formar pessoas e criar uma área no Brasil. E sequenciar o genoma. Sequenciar, nós sequenciamos, saiu na Nature, está lá. A outra parte, eu ainda não vi os resultados. Eu converso com muita gente, as pessoas falam, para mim foi importante, não, eu mudei minha vida, eu fiz isso, fiz aquilo. Mas o impacto na comunidade de São Paulo, eu nunca vi um trabalho mesmo de levantamento de história da ciência para ver qual foi o real impacto, né? Quer dizer, ou pensar o que teria acontecido se a Xilela não tivesse existido, como é que teria desenvolvido a biologia molecular no estado de São Paulo, né? Então, eu acho que a gente deve um pouco isso ao objetivo, um dos principais objetivos da época, né? Então, era isso que eu queria dizer. É uma delícia vir aqui encontrar um monte de gente conhecida, que eu não vejo faz tempo, e lembrar das reuniões que a gente tinha nessa sala aqui. Então, foi muito legal. E, para mim, só resta agradecer todo mundo que fez isso junto, porque foram anos muito divertidos. Obrigado.
Muito obrigado, professor Fernando. Pedir a gentileza, então, professor Zago, professor Mello e Fernando, que se acomodem na plateia. Convidar a professora Mariane Vens-Luz, da USP da FAPESP, para coordenar o evento a partir de agora.
Hello, everyone. Ana Maria and myself, we are very pleased to have you all here. It's a great moment. We have been organizing this meeting for 3 years already, so we weren't aware of the COVID pandemic when we started all this, right? And I wish you were here by my side, you know. On the other side, I would also like to thank all the speakers because they had to switch their agenda 3 times in order for us to be here today. And we are now starting a new wave of COVID-19, so I would ask everyone to keep their mask on. I will not repeat myself regarding all the presentations we had before, but I would like to stress the organization of this meeting. We tried to organize it in 3 large sectors, one of which is the first one in this morning. It will address the genomics of pathogens. In the afternoon, we will discuss the agronomic and environmental genomics with different views. And tomorrow, as Professor Zago mentioned, we will address the cancer genomics, which I think it's very important, these 3, because they represent major areas of Brazilian research activity, not only in the state of São Paulo, but also in Brazil. And I would like to say that the current research projects that are ongoing right now, they have benefited by all the initial investment. Although we don't have the evaluation, as you mentioned, Fernando, we will see several of young researchers that were either PhD students or early career postdocs that will be presenting their research here. but I agree with you, it would be great to prepare a large document on that. It is important also to say that this initial investment was very important for the avenues that genomics has taken us through science, technology, and innovation, and also to help us address several of the societal problems that are well summarized in the 17 Sustainable Development Goals. Genomics is everywhere, okay? So with this, I will start the presentation of the first session, and I will be very, very happy to invite Professor José Fernando Paris. He is an electronic engineer by training, and I thought you were a physicist. Yeah. Oh, great. But most of all, he was our scientific director at FAPESP, who really took the chance to make this a game change. He was the scientific director from 1993 to 2005. He has challenged the research agenda by introducing the concept of research program initiated with UNSA and further followed with all other great programs that FAPESP has developed. He is a member of the Brazilian Academy of Science and atuais. As a result of his initiative, he was granted the National Gran Cruz Ordem do Mérito Científico Tecnológico from Brazil, and he is currently the president of Recepta Biopharma, one of the spin-offs of the UNSA program. With a great pleasure, I invite you, Paris, to give your presentation. Thank you so much.
Good morning, everybody. Even Saturday night, I was in doubt whether I would be able to. Even Saturday night was in doubt whether I would be able to be here today, because as you can see from my voice, and probably you notice by my coughing all the time, I mean, I'm still recovering from a very bad cold, not COVID. I mean, I tested negative yesterday, so be reassured, I'm not. And first I want to thank you, for these very kind words that were said by my friend Luis. And I am very proud and honored by this invitation, and very happy that we are commemorating this 20+2. First, I think there is a misname here, because it should be 20+5. Because everything started in 1997. And that's when we took these decisions, when the idea came out. Because I don't have a presentation. Actually, most of what I'm going to say here has been said by Zago and Luiz Eugênio and Fernando, also by Marianne, but I think this is a contribution for this effort of having organized the history of this, a research project with documents, because I have been warned by André Goffo, who was one of the advisors of this project. He said, well, when you tell the history of this project, it should not be an oral history, it should be documented with the emails that were exchanged at the time. This does exist. And we should try to recover that, to have, a research project, from a historic point of view, not just by oral deposits. Because he was telling me that this, what happened in Europe, when they told the history of the Saccharomyces cerevisiae project. There were claims that were completely out of because the memory betrays everybody. So this is an important fact. And I think that this effort should go in that direction that Fernando mentioned before. It should make a consistent evaluation of the impact of this project. So this all began in, not May 1st, 97, before that. Because, I was scientific director at the time, and I, as a physicist and electronic engineer, I was listening to the buzz made by biotech. Biotech here, biotech there, and I came to Fernando, who was one of my advisors, and said, Fernando, we need something daring, something big in biotechnology. And Fernando is interesting. He's the ideal person for you to tease and challenge, because he doesn't give up. He lives up to your expectation. So I kept that message. We have to do something big. And then May 1st, this was it's a Brazilian holiday, Workers' Day, not in the US though, but in Brazil it is a national holiday, Workers' Day. And I was at the beach, and I got a phone call from Fernando. Oh, I had an idea. And he explained to me, I could barely understand as a physicist what sequencing a genome would be. I learned quickly that, because genome sequence is something that a physicist can understand. And I think physicists like genomics more than biologists. That's what I learned at that time. I was fascinated by the idea. And the point is that he said, well, why don't you come to my place? A country home he has in Piracaya, a beautiful place. And we spent the rest of the weekend, extended weekend, to discuss this idea. I was fascinated by the idea. And interesting, all the elements were already in the basic idea that he proposed. A network, the choice of the organism, a training, and the cost, estimated cost. I was really fascinated by the idea. And I had the standard reaction I had at that time because lots of people came with the beautiful ideas. This is the beauty of being scientific director. You get an input from the community. And this is one of the cases. And I told Fernando, please, write it down. And he did it. He prepared a summary of the idea, a pre-project, and very consistent with I was very happy. And then I summoned a meeting with leading scientists from all areas that are involved with genetics. I don't remember the names of everybody, but Paula Ruda was certainly there. First, João Lúcio Azevedo, Marco Antônio from Fundo Citrus, Mariana Zatz was also there. So we had all, and we shared the idea. And there was a unanimous reaction. Yes, that's a great idea. I was concerned because this was a project that should go around $15 million. This was, at that time, imagine even now, one of the biggest research projects from the costs involved. I was scared because, so unanimity scares you because, very often unanimity is in the wrong side. So I said, well, okay, let's move forward. And then we decided that we needed an international feedback on that. That's important. We invited André Goffo, and I think Paula Ruda was involved in getting him involved. Thank you. That was a great contribution. And André Goffo had been the DNA coordinator of a similar project for the Saccharomyces cerevisiae that was conducted in Europe, a network of labs. So the idea of a network was pre-existence to them. And then we invited also Steve Oliver. And this was a bit confusing because how did we get Steve Oliver? We had a meeting here in this room, launching the program of small business, the same very same year, 1997, and we invited the representatives of the British Council in São Paulo. And then I was talking to him, and my style is to share an idea. I told him, look, I need the best specialists in genomics from the UK. And he said, I have the name. Immediately, he said, in this corridor here, I have the name, Steve Oliver. He was the coordinator of the Saccharomyces cerevisiae. I said, oh my God, it's the same. Oliver, go for, no, he was indeed the coordinator in the UK. And we invited him. Both came, and the surprise is that they unanimously found it was a great idea. Great idea. So I was more reassured. I mean, let's move forward. When Fernando mentioned the project, so well, we should choose an organism that's related to the agriculture. That was in the very initial idea. So in these initial meetings, the idea of Xylella fastidiosa appeared. I mean, it was already on the table. But there was also clear understanding that it was impossible. Actually, fastidiosa is not, a poetic name. It means that the culture grows very slowly, even in the plant. And so nobody here knew how to grow the culture of this bacteria, which was needed for having a library for having the project. So it was discarded. And actually, this was never, this is important, this was never a project for the Xylella fastidiosa. This was a project for genome, a capacity building project. This was a big misconception, even among scientists that didn't understand that what we had in mind was not getting the genome of the Xylella, was a capacity building. So Xylella was excluded at that time. But the consultants go for Steve Oliver proposed 3 criteria for choosing the organism. The size of the genome should be commensurate with the capacity that we had. It should not be a very big genome. Should not have to be too small, like a virus, that would be not justifying the creation of a network. Should be of scientific relevance, should stimulate researchers to analyze the genome. And latest, but of socio-economic relevance. These meetings, at some moment, this is a very important moment. Goffo and Oliver told us there will be a bottleneck in the project. You don't have bioinformatics in Brazil for that. And they offered us some collaboration with some German center that would do the bioinformatics. But then, all of a sudden, we had in the meetings 2 new players, João Setúbal and João Meidanes, which I didn't know before. They appeared, and I was told that they were excellent researchers, and they would be interested in doing the bioinformatics. I didn't know how good they were. I must say, I was not aware of that. Actually, I learned afterwards that they had written a book on bioinformatics, a book that was even being used at MIT. And if you look at Amazon, it was one of, if not the first book on bioinformatics. And they were in a meeting. Then I remember that, I think it was João Setúbal said, well, Professor, it's a dream come true to work with a real genome, not a simulated genomics, because that's what we're doing. I think I heard these words at that time. So that's something that surprises you. The excellence that exists in our system here. Of course, our system is big. Its excellence is measured by the number of islands we have, unfortunately. But we have these islands. And it's important, it's very important to explore the potential that we have there to do daring projects. This is how they entered the game. João Setúbal, João Meidanes, the 2 Joãos. There was another third João that joined later on, but these were the Joãos that told us that we had bioinformatics. So, and then I had a meeting at lunchtime with Ricardo Brentani. Brentani wanted to talk about some problems of the graduate course school at the hospital Câncer. And during this, I shared the idea. Look, we are preparing a genome project. And so he was very enthusiastic about it. Oh, that's great. That's great. I will bring you 2 people that you love to have in your team. That's how. Then he suggested inviting Andrew Simpson and Joaquim Machado. I didn't know neither of them. So we invited them to participate in it. And Andy entered with his standard enthusiasm. I mean, I had a quick read in his vita, and I realized he was an important scientist. And he was very enthusiastic about it. And he's a very seductive person. He got a lot of, very welcomed by the group, very welcomed. And he was enthusiastic. He wanted to press us to make a decision on the organism. He put a lot of pressure. And then there was I was very concerned. I was present every single meeting because, this was a very big project. It was such a big responsibility. At some moment, I would have to take the ideas to the consideration of the FAPESP Board of Trustees. So I had to understand at least the main ideas. And then but we had a visit from 2 eminent Brazilian scientists from New York, Ruth and Victor Nussensweig, were visiting FAPESP. And I have to withdraw from the meeting and have this interview with them. When I came back, they told me, we have the organism. The organism should be Thiobacillus ferroxidans. I'm introducing this guy to you. I was impressed. They had a lot of arguments for that. It's very important. It's a bacterium. Size was actually, this organism was sequenced afterwards by another center. I think it was Tiger that sequenced the Thiobacillus. And it's important in environment for remediation of waters. There was an interest of Vale do Rio Doce in this project. So I thought, fulfills all the. But then, almost decided. So it was not going to be so. But then I got a phone call. Next slide, please. It's here. Then I got a phone call from the Fundo Citrus. Ademerval Garcia. He was the president of Fundo Citrus. And he said, we are very enthusiastic about this project. We support the idea of being acelera. And I told him, the problem is that we didn't know how to grow its culture. No, but I know who can do that. He had invited me. He had invited Professor Joseph Bové. Joseph Bové is probably more well-known. He is an important biochemist. His son is very famous. He was candidate to president. Very crazy guy, I would say. I had an interview with French people here, and they said, we invited Professor Bové, le sage, pas le fou. Anyway, and on a Saturday afternoon, we had a meeting here at FAPESP with Professor Bové and the whole group. And Professor Bové, he knew a lot about the bacteria because he had proven the causal relationship between Xylella and the amarelinho, the disease that it provoked. There was this so-called Pasteur's postulates. Actually, there was another paper, an American US paper, proving this causal relationship, which is very critical. And I will come later to that. So I listened to that. I was impressed because Professor Bouvet had a passionate and convincing defense of me, and I know how to grow this culture. I have done that because when I studied this, I know how to. Culture growing is something tricky. I mean, for a physicist, it's very difficult to understand that some things are not so deterministic. It depends on the mustache of the. It's not. I mean, it's a different science. It's like crystal growing. It's just the same thing. But it was very convincing. He had this real experience. So he made this defense. I mean, and Fundo was prepared to collaborate with us, with some small costs that would. And when they, we had after the meeting, I had discussed internally the case. Say, well, how do you think? And everybody it was unanimous. Well, let's go for celebrará. That's the moment we decided for the celebrará. So this was never a project for celebrará. It was an opportunistic bacterium for this project. This is an important consideration. So we are prepared. At that moment, we had to get approval. I mean, the scientific director, in general, has sufficient this capacity to take decisions about supporting a project, but not a project of this order of magnitude. So I had to take it to the Board of Trustees. And we prepared that, and I must say it was not a unanimous decision by the board. It was approved unanimously, but there was some resistance. Well, that's risky. This is a terrible word. Risky. This word makes me ill to listen to the risk. These are the good projects with risk-taking. Our scientific community is very much averse to risk-taking. This is an important fact. And so there's this risk. Actually, there was one board member said, well, let's vote. Because, everything in this board of trustees is decided by consensus. There is never a vote. There is never a vote. So when this board members proposed a vote, it was unanimously approved. But this was an important step to require a vote at that time. Otherwise, there would be a problem here. But there was a political dimension also here to this project. Not only internally, also outside in the community at large. That's very important. I got a letter from a very respected, a great scientist, one of the leaders of biochemistry in Brazil, who sent me a letter. So look, this project doesn't make any sense at all. Doesn't make any sense at all. First of all, what are you doing with if you knew the genome of the xylella? What are you going with that? Do with that? Second, nobody knows whether this bacteria is really responsible for the disease. And third, I heard rumors that this project was responding to some political internal crisis of FAPESP that was trying to address. So, first, my response was very easy because I say, Professor, with all due respect, because this was an important leadership, and this was an opinion shared by all the community, the leaders. This is important. Max Planck, I will say something very politically incorrect, but these are not my words. These are Max Planck's words. Please forgive me in advance. With due respect to all old scientists and good scientists. Because Max Planck was leading, one of the inventors of quantum mechanics. He said, science progress also because the good scientists die. And with all due respect to the good scientists, this is a very respect, but this is the anthropology of science, sociology of science, because good scientists have such a political power. Their word is very important. And I would say the following, because since I was a physicist, I'm a physicist, I used to be a physicist. And if I heard, this criticism come from one of my advisors in physics, I would probably be very concerned. But since I was not a physicist, I was respecting the opinion of the young people that were with me. People that, and if you look at the people that participate in the project, mostly young people. This is a very important fact. The old generation stayed away from the project. This is something to be analyzed. Some criticizing, the others scared. And some later on had this, what Andrew Simpson said, there is this oh shit syndrome. So, anyway, this is the truth. It's important that you understand this context. And so we decided on this structure, a DNA coordinator. Andrew Simpson was a very natural choice to work on it, because Andy is a natural leader for that. He knows how to combine pressure with the stimulus. It was a very diverse group that was there. Most people were not trained in genome sequencing. And that's why we created also these two training centers. One with Fernando Reinach at USP, the other with Paula Ruda at Unicamp. That would be training these people. This is like capacity building. Actually, I didn't complete my answer to this distinguished professor who sent me the letter. So there are two independent proofs that there is this causal relationship. Second, this is not the project of Celera. This is a capacity building project, very much like a project that was funded by FAPESP, a very important project that created biochemistry in São Paulo. And he was one of the leaders. And third, the mention to a political crisis doesn't make any sense at all, and I'm not going to elaborate on that. Later on, he apologized to me. He apologized. He was a serious scientist, but he had sent a letter to a COP with 10 or 20 people. So he disseminated his discontent with the project. This was political heavy, because everywhere I went, well, what are we doing with this project? You are spending all the money of FAPESP. This was completely untrue. At that time, FAPESP, I don't know how much time I have left. I have to... Anyway, so we decided we selected 34 centers, and we had a collaboration with Bordeaux. And Joaquim Machado, one of these guys at the Brentani, proposed to him, he went to Bordeaux, and he was generating the first library that was used. And actually, this library was important, because this was the gold standard for the library, because it was the library that, of the strain, for which he had proved the causal relationship. And there was not another strain that we would never know whether it was the one responsible for the disease. So the library was generated at Bordeaux. Joaquim Machado. And we got, this reaction of the community that I already mentioned. So, and then we got the Sussex, 2 years later. Actually, we started in 1998. And in the 2000, the project was completed. Fernando said we had a lot of fun. Fun for him. Was a lot of anguish for me. I regret to get that done. This project is not just do it. Bioinformatics is very complicated here. A lot of repetitions of this. This was a challenge. We even invited the people from Washington University to see whether we are doing things properly. They are concerned about that. No, they came here and said, well, everything is right. This is a standard challenge, these repetitions. Have to be faced. So there was a lot of tension. I was always believing that, but anyway. So it was completed, it was published by Nature. It deserves not just a publication, was an editorial. Later published, and it had an editorial, and then another article in News and Views analyzing the project. It was very, 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 very publication. Thank you. And then, and almost before we had completed the project, we started another project that was proposed by the Ludwig Institute for Cancer Research. Ludwig Institute is a not-for-profit institution, US-based, that used to have a branch in Brazil. And this branch, director of this branch was Ricardo Brintani. So Ludwig was already a partner of the project because in a very visionary way, Ludwig is dedicated to cancer. What the hell was Andrew Simpson coordinating a project of a plant pathogen? So this shows that there was a misunderstanding that this was not a project of Salela. This was a capacity building, and this capacity that was being created was going to be used in other projects, could be used in other projects. And they immediately came with the idea of studying expressed genes in tumor cells. This was not a genome. At some stage, Brazil was the second country in the number of human ESTs in public. Here, I think we didn't do as well as we should have done. Because we decided at that time, because there was a big competition with two genomes, Craig Venter's genome and the public genome. They are competing. And Craig Venter was demonized by the scientific community because he wanted to make money out of it. And there was a concern that we should share the data in a generous way, because otherwise we would be aligning with Craig Venter. I think we could have done better than that. We should have shared the data, but we should have got the credit that we deserved, because our results are used by both efforts, the public and Craig Venter's for the sequencing. It's a lot of information. We're the second country information for our public. Well, tumor cells, but they don't matter. I mean, they are human cells. So there were a lot of publications, as Zago presented here. I mean, it was a scientific success. But I think we didn't get, FAPESP didn't get, the project didn't get the credit it should have got. And then we had a lot of other projects. And most surprising, we had a commissioning from the US Department of Agriculture. That was incredible. They want us to sequence the other strain of Xylella fastidiosa that was responsible for Pierce's disease that was attacking the vineyards in California. I got a phone call from a journalist from the San Francisco Chronicle. He said, Professor, what do you say? There was an institute in California criticizing the US Department of Agriculture for commissioning it in Brazil. Because they said they could do it faster, better, and for free. And I was asked by the journalist of the San Francisco Chronicle, Professor, what do you have to say about that? Well, faster and better, I doubt. For free is none of my business. So we did it here. And this, I tried to find yesterday night, but I couldn't find. There's some beautiful reaction from the school teachers in California. They asked the students to thank Brazilian scientists for studying the genome of Xylella for the vineyards. This was incredible. This was unheard of. So this is very, very beautiful. So then we had all this. I mean, I think the meaning experience also the success was. So the infrastructure, physical and human qualification that allowed us to do a lot of other projects. This is important. And then, well, of course, we had this nature, cover page and so on. Actually, the cover, there was this interesting, how did we get the cover? Somehow, Governor Covas learned about that it was going to be published in Nature. And he said, that there was going to be cover. I said, my God, we have to honor that. Then we learned that the choice of the cover is not based on scientific consideration. is decided by a photographer at Nature, who chooses where he has beautiful pictures. Then we say, well, send them a lot of pictures. So these pictures were sent by us, so that he was seduced by the pictures, and that's why we made the cover. That's the truth. It's a decision by a photographer. So this is the New York Times. That's it. New York. I mean, actually, I'm very proud of that, because this was published with a big picture of mine. This is the one I like best, let's say. I spared you with a picture. So, the New York Times, I mean, almost a year afterwards. This is Claire Fraser. Claire Fraser was the wife of Craig Venter, and they were together at Tiger. Tiger, it was the Institute for Genomic Research. Actually, the name ONSA was a game of words here, I mean, because onça is a jaguar in Portuguese, with a different spelling, very much like tiger, or there is an E missing, so there's also misspelling here. So the onça was the response to, Brazilian response to the tiger. Actually, we had a visit to Craig Venter when we told him about our project, and he wished us luck so that we could not bring this repute to the felines. So, this is Claire Fraser, the president of Tiger. They have really become recognized as an important player in the field, making a major contribution to the international, I mean, science of the highest quality, comparable to anything that's being done in the large sequence in the US or Europe. Le Figaro. Le Figaro, le Brésil est parmi les grands. Annonce de députada. And then, that's the article that Zago was quoting before. 10 years later, Nature had an editorial celebrating 10 years of the publishing of the cellular genome. And in boldface here, perhaps more than anything like that, the benefits of aiming high. That's the lesson. Still true. Scientists undertook a major project executed with precision and published. The results were broadcast by mainstream. Parents believe that this singular outcome even helped to change Brazilian science relationship with the Brazilian media. That's true. We changed the relationship with the Brazilian press here. I mean, the coverage of science in Brazil was very scarce, very rare. And after that, I mean, we saw the reporters and the press coming to us asking for news, and this changed the relationship. There was a qualitative change of the relationship. I think I missed something important here. This one I like best. Samba, football, and genomics. Look at that. The list of things for which Brazil, this is The Economist. The list of things for which Brazil is renowned has suddenly got longer. Only America and Britain have more than that number of human STs identified. At that time, we were third. Later, we became second. So, that's it. That's the history, I mean, as I recall. And I think we should make now something more systematic. As historical researchers, someone done history of science research, to have this with documented history, not these oral recollections that we shared here today, but and we have to move forward. We have to do more daring science. I mean, I think, especially in São Paulo, the scientific community is in a mood of not facing challenge. I mean, the word risk, this is a risk-averse community, yes, risk-averse. And so that's where FAPESP enters, because FAPESP can take this risk. and and this is what happened. And I look forward for more of that happening in the future. Thank you.
Pérez, thank you so much for setting the scene. The idea of the meeting is we will have a 40-minute presentation setting the scene at the time. Then we will have 3 presentations that will present the nowadays research that are ongoing. And then we will have a later presentation that will address what are the future and the challenges in the area. So with this, I will invite the next speaker, Dr. Alessandra Alves de Souza. She is a biology and currently a research associate at the Instituto Agronômico de Campinas. Her initial steps in phytopathology were undertaken during the Xylella project. She was a PhD student, right? Wow. So she was a master's student at that time. Nowadays, she is recognized as a leading scientist in citrus disease, having received multiple recognitions awards from Bunge Young Investigator in 2005, BASF Top Scientist in 2015, and 2018 she was recognized by her contributions in citrus pathogens issued by the American Society of Phytopathology. Please, Alessandra, come join the floor and tell us the story about Xylella.
So I'm a little bit nervous because I feel like 27 years looking for you guys over here, Fernando and Paula Ruda, everybody. So thank you, Marie, for the invitation. It's a really pleasure for me to be here. And so thank you, FAPESP organizer, for invitation, for giving me the opportunity to show a little bit, to talk a little bit about genomics on here. Let me see how it works. The pointer? Even the pointer? Oh, sorry. Okay, let's try without the pointer. Okay, so I'm going to talk about the genomics on pathogen and focus on Xylella fastidiosa. So I'm going to try to speak in 20 minutes what happened in 22 years regarding the impact of this project on the scientific community, a little bit about my story as a consequence of the Genome Project, and also the research we did on Xylella fastidiosa in my institution, that is Citrus Research Center, that belongs to Agronomic Institute, the institute where I work. All right, so as it was already said by everybody, by Dr. Pérez and also by Dr. Zago, so this project was a strategic vision of FAPESP to promote the development of molecular biology and biotechnology, and to training of human resource, and to have a multiplier effect, because a lot of students was working on this project, including myself in that time, and so to have an international scientific impact. So it was the main goals of this project, and so the genome sequence, as you already know, and the choice of Xylella fastidiosa. But what also is important to mention that Xylella fastidiosa was also chosen because citrus is very important to São Paulo state. All right, so The citrus culture bring in that time, today is more, around $1.5 billion a year to Brazilian agribusiness. And São Paulo is responsible for more than 8% of this production. And so in that time, this disease that we named amarelinho in Portuguese, or citrus variegated chlorosis, CVC, it was causing a very large damage in the citrus culture. And just to have an idea, in 1997, if you see over there, we have more than 30% of the orchards with the incidence of the disease. So it was a very problem for our citrus culture, and very little was known about this bacteria. So that's why it was chosen, because the importance of São Paulo State in agriculture, and nothing was known, or a little bit was known about this bacteria. And so it was already said, but I'm going to explain it very fast here. And then it was constructed, this organizer with the steering committee, DNA laboratories that Paula Ruda was in charge with this lab, and sequence labs. And the sequence labs, if you observe over there, Corderópolis, or here, is the lab where I worked at that time. And we was one of these sequence labs. And then the bioinformatics, that was with Setúbal, Meidanes, and also Kitajima in that time, that helped it a lot during the sequence genome. And so this was the team in Corderópolis, Marcos Machado, who was our PI and coordinator, Elvésio Della Coleta. So we grow Xylella in our lab. And for this reason, the bacteria that came from France go to our lab, and Elvésio grew the bacteria and distributed the bacteria to the DNA lab. So we were the only lab that could grow the bacteria in that time. And so Elvésio distribute this, and then DNA, they come back, and this is the team responsible for the sequence team. So Maria Angeliane, Maria Luisa, and myself. And when the sequence finished, we started another step. That was the annotation of the genes in this genome. That was a very challenge for us too. And then Takita and I was the team, composed the team involved with the genome annotation. So here is one point very interesting, because I'd like to mention that this bacteria was very curious, because we was responsible to annotate this, pathogenicity and adaptation category, because we work on citrus disease. And so we were responsible to annotate genes involved with this category. However, we were looking for this model, classic model, that is the type 3 sequestration system, because all the bacteria that caused plant disease in that time used this type 3 sequestration system to cause disease in plants. And so we didn't find any genes associated with this sequestration system. And then everybody, including Aranha, that is over there, started to looking for these genes. And there is these genes are secreted this effector, that now the name is effector, but before it was named AVR proteins. And this protein can manipulate the plant cell and then cause symptoms. But nobody found any genes in Xylella related with this system. That was very known for the other bacteria that cause plant disease in that time. And so it was a highlight of the paper, that there is absence of these genes in Xylella, so Xylella was indeed very different, and the highlight in the end of the text was that probably a different mechanism not involved with AVR protein could be associated with this bacteria. And now, in relation to the initial goals, we can mention that, yes, the FAPESP has a very success that Dr. Perez already explained you, because we published the paper in the Nature, was covered. Also, we was invited by a researcher from the US to sequence the Xylella that causes Pierce disease, that Marianne was the coordinator of this project. and I participated too. That was very nice for us. And yes, we have a training of human resource and multiply effect. A lot of research was involved in the paper, and also because of this program, the Xylella genome, the CNPq start a program to sequence other organisms. And then we multiply the effect to our country, because there are a lot of other labs in Brazil that take money from CNPq to start sequence organisms. And the other, as was already said, other organisms by FAPESP, CNPq, and another FAPs around our country, around Brazil. And so again, indeed, it was a very nice publication. The Economist journal already showed by Dr. Pérez that Brazil is not samba or football. We also have genomes. It was very celebrated by the community. And then, as I told you, in 2000, before 2000, very little is known about Xylella. And now, if you put the name Xylella fastidiosa only in the PubMed database, you can see the increase of knowledge after the genome sequence. But, okay, the first goals was reached. Training people, multiply effect, and now we have a problem. We have the CVC caused disease in citrus. How to solve this problem? If the mechanism of pathogenesis is not type 3 sequencing system, what is this? And so the FAPESP just immediately after the genome sequence opened another project that was the Xylella fastidiosa functional genomics to try to understand the function of the genes of the genome, and also what is the mechanism of pathogenesis of this bacteria and how to control this bacteria. And so we were one of them, that's IAC, the Agronomic Institute. The project was developed by us. And then the idea was to identify genes that could be associated with the pathogenicity. So here I'd like to point some important steps in my professional life, because the functional genome of Xylella fastidiosa, this project was part of my PhD that I developed at the Unicamp. And then I got my first job in Embrapa. It was in 2003. And in 2005, I left Embrapa and I went to Agronomic Institute as a scientific research. So it was a very important in my career. And so what we found with this project was the pathogenicity of Xylella fastidiosa is associated with the ability of bacteria to move and colonize, forming biofilm in the xylem vessels. But this biofilm was quite similar with the bacteria that cause human disease. The genes and proteins that encoded by such genes is very close with the bacteria that cause human disease, especially disease that in respiratory disease. And this found, due these results, I got all very important achievements in that busted my career, because in 2005 I got this award from Bung Foundation that he said. 2005, this other one, the young geneticist by Brazilian Genetic Society. And in 2005, I got my first project that was a younger scientist by FAPESP. And then it opens door for me to start as a professor, visiting professor, and try into the genome molecular biology postgraduate program in the Unicamp. And after that, Fernando, the multiply effect was even more. Only with me, that one of the hundreds of people that participate of the project in that time. And after that, I could have much more students. And all of them, this was just example of some of them, but there are much more. And all of them was training in molecular biology and biotechnology. And then some of them are in the university now, in a private company. Some of them also not in Brazil anymore. But all of them trained in molecular biology and biotechnology. And so what we studied was, in general, what my student was involved was to understand the plant-microbe interaction to try to find key genes to be used aiming pathogen control. So we studied the plant side and also the bacterial side, because not only the bacteria, but there are plant-pathogen interactions. So which genes could be used to try to apply, and then we have the control. So I'm going to show you here just a few examples, some highlights that we found. One of them, the highlight, another point that's very important to mention is that the Genome project opened doors for us to have international collaboration in different universities around the world. And so it was good to improve the impact of our research. And one of them we found was the one gene in Xylella fastidiosa that is responsible the bacteria do not move in the plant. And so we took this gene from the bacteria and put this gene in the plant. It was very nice. We have a cover on this paper. And also, as the FAPESP, we have the genome of Xanthomonas citri that causes citrus disease. We started to study genes that could be involved in both bacteria and in citrus plants to use some technology that could be applied to have resistance to both bacteria. And so this one that was published, we have a very nice repercussion on the media, included the FAPESP magazine. And another one is one gene from tangerine, because tangerine and sweet orange are naturally crossed. So we start a process that could be natural, but was more fast when we took one gene to transform to another one. And they also have very good repercussion in the media. We have collaborated in the UC Davis, the other one in UC Berkeley, and this one is another highlight. Another highlight that is EFR in collaboration with the people from TSL. It was now a good repercussion too. These plants are now on the field condition, and we are testing them because the development of the plant and also the quality of the fruits. And another one that I'd like to highlight here, because it's very nice, is because when we try to understand which genes and protein was involved in the Xylella pathogenicity, we observe that these proteins that form biofilm use the disulfide bond to do this biofilm. And so, and use very similar to bacteria that cause human disease. And so we start to looking for what is used in human that you can use in agriculture, but it should be cheaper and so sustainable. And then we verified this one, N-acetylcysteine, because the cysteine analog that is used to break the sulfide bond, and it also has an antioxidant effect. And we did a lot of work. We published some paper. We got a patent in 2011. It was considered in 2018, and now it's submitted to US too. And what do we have? So we got a lot of results that this molecule could be applied in the field condition, but how to transform in the product to reach the farmer. It was very difficult when you have something good in the university or in academy, but how to put it in the field condition, how to produce a large scale. It was it is something to be improved in our country. And so, FAPESP, thank you again. My ex-postdoc opened a spin-off, and then she got the PP1, 2, and now it's with 3. And so the results in the field condition was very nice. Now this CIACAMP, that's the name of the spin-off, had more employers now. And because the good results, this other one, Amazon AgroScience, that is a private company, put money in the spin-off, and now we have 3 products that is already available, and not only for Xylella, but to other pathogen that could be this molecule could also break this interaction with the plant, and they also improve the plant response because it is an antioxidant molecule. And this is a very good example as the research, basic research can be applied, and also the royalties that came into our institution goes to the research to move again the scientific research to try to solve problems. And so how is the situation of CVC nowadays? Everybody can ask that. Okay, a lot of money, but this is the situation. We have no CVC problem in citrus in São Paulo anymore. I can explain you 1 hour here to explain how it occurs, but I don't have time, but I'm open for question after. But in Xylella, we have a problem. I think that Dr. Zago already said. Xylella is still a problem in the world. This was a publication in 2019 by Phytopathology only about the address a new global threat of Xylella fastidiosa, because it causes disease in many different crops, and is a very hard problem in olive in the south of Italy, in Europe. This is the situation. You can see the name of the disease, olive quick decline, caused by Xylella fastidiosa in Puglia. It's a very dangerous problem. And so, just to show you the importance of the investments in the Xylella project, we are a reference here. Because in 2013, this disease was found in Italy, but nobody was able to isolate the bacteria and grow this bacteria in the lab. So the Maria Saponari from Italy came into our lab to learn with Elvésio Della Coletta, he's over there, and how to isolate the bacteria and how to grow this bacteria. And after that, she came back to the Italy, and then she could prove the Koch postulate that Xylella were indeed the agent causal of this disease. And a lot of research improved again related to Xylella fastidiosa. So I almost finished my talk. And just to show you the importance, in the beginning, we did nothing. Now we are a reference. And we are helping collaborate with the people abroad in Europe. Brazil is a protagonist in basic and applied knowledge involving Xylella. And so we have this project with the European Commission that is Horizon 2020. The first one, Point and XF actors, was already finished. And now we are going another one that Bexil, that is also supported by European Commission, where we are collaborating with them. We have a lot of meetings. These people are very involved. I don't know if you remember, but the Purcell and the Hoppings, Steve Lindau, people that work on Xylella for a long time in the US, and they also are involved with this team. And we are together to try to help people from Italy to solve or to deal with this disease. And to finish my talk, I would like to emphasize that we have a good example of Xylella fastidiosa that's from basic to applied knowledge, but it indeed takes time. But I'd like just to take a message that without basic and fundamental research, we'll never have applied knowledge or technology transfer. It takes time, but it's necessary to have science, good science, that when we have this applied knowledge, we know why. And so thank you very much, FAPESP, for believing it. And so I'd like to thank my team, especially Marcos Machado, who was my PI in that time, and believe in myself to work on this Xylella project. I was only a master student at that time, and then I did my PhD, and all my career work on this bacteria. Thank you very much for all of you.
Thank you, Alessandra. Very inspiring presentation and showing how the investment is important. We will now move to a different pathogen. I would like to invite Jorge Elias Calil Filho. He is a medical doctor specialized in allergy and immunity, president of the Instituto Todos pela Ciência, which with an important role nationwide in building biotechnology in Brazil. He is a member of the Brazilian Academy of Science. His scientific contribution in the field of immunogenetics has granted him the Ordem da Gran Cruz in Brazil and Chevalier de l'Ordre National du Mérite in France. And I would like to invite you to give your presentation on viruses and immunity. Thank you so much, George.
Thank you very much for your presentation. Thank you, Marianne. Thank you, Ana Maria. Marianne, Ana Maria, it's a good double. Thank you for. Okay, sorry. It's my pleasure also to meet old chaps that I don't see for a long time, like Paulo Arruda in the biotechs, and also Fernando Reinach, my good friend since the time we were Howard Hughes Medical Institute. Well, FAPESP did a great job for sure, and I think it's very important to dare in science. And I think this is a problem that we have in our science. It has been said by other people that were here on the stage before me, but I think that very often we don't dare. We try to do what we're supposed to do. I think we are pretty much conservative. And what has been done here with all these people that are so important for Brazilian science was very important. It was a turning point in the science in São Paulo. And this is spread, of course, to Brazil. And it became very important, of course. What I'm trying to review today is how did we perform in the genomic surveillance of SARS-CoV-2? This presentation should be done by Esther Sabino, that's sitting here, but she was supposed to be in Paris, but she gave up. She's here. And she was the one that helped me a lot to prepare this presentation. So how did we perform during genomics? Because with all this effort of doing genomics in Brazil, in São Paulo before and then in Brazil, did we maintain? Could we invest in order to be ready? What about our readiness for pandemics? How long did you take to discover that Zika was important in Brazil? How much do we actually follow malaria? How much do we follow dengue and so on at the genomic level? I mean, do we know if there is, what about the diversity of the virus that are spreading? And how important is this to control the pandemics? I mean, we've seen that it was very important. And were we actually prepared for that? So sorry if I tell you things that are obvious to you, but when we think in health and how we should be able to do surveillance of disease, we have to be ready to do metagenomics of samples from different areas in order to see what's going on, or what kind of virus do we have, or even bacteria that are spread in the country. But once we have we have identified the pathogen, and it took some time, as I mentioned to you, to realize that Zika was important in the country. We have to be able to doing targeted sequencing and follow up what's going on with a particular pandemics. I mentioned to you, we're going to talk about SARS-CoV-2, but what about the other diseases? And also, we have to follow the population sequencing and see exactly how this deviation of the pathogen could occur and how important could be clinically, and how it could be important also to escape vaccination profiles and so on. So it's very important for such a big country like Brazil, and we have to be prepared. So when we look, and this is this was done by Anderson Brito that works, it's one of my collaborators at the Instituto Todos pela Saúde. We have seen that there are many disparities in the global genomic surveillance of the disease. Don't have the time to go in detail to this map here, and the pointer doesn't work. You know, the red pointer doesn't work on screens. You need to buy here FAPESP a green one. I see. But I mean, if you can see. Europe was able to follow it, and mainly in UK, they could follow the sequences very well, and they could identify the different variants. Same occurred also with part of South Africa, as you remember. But when you look at the bottom of the table, it's where Latin American is, and you can see a lot of orange and stuff. So this means that we were unable to do the sequencing, so we could not follow. And this, as you can see, by mid 2021, we started doing much more sequencing. So the technology was there, the investment was not. And I think that the politicians did not realize how important it was to follow disease. It took some time, perhaps 1 year, in order to start it. So this, again, it's done by Anderson Brito from the... and then you can calculate, I'm not going to go in detail of this slide, but you can calculate what are the chances that you have to pick up a variant if you do sequencing, and the percentage of the actual cases that you can sequence, and the time that you take to sequence. And this is all presented, you can see in this paper from Anderson Brito. What are the chances of picking up a variant? So we didn't have the chances of picking up a variant, and that's why Japan took this variant that was typical Brazilian, the gamma variant, right? And then after that, we were able to do it. But if we could sequence more and quicker, we would be able to figure out what were the variants that were started in Brazil, and perhaps they do something. So here, as you may remember, at the beginning of the pandemics, the Itaú Bank decided to give $1 billion for helping in the control of these of the pandemics, and then other companies also helped, and the total was something like $1.3 billion of money. And this money was spent in the early beginning to buy masks and alcohol and so on, and then you figure out they could not go very far with this approach and this strategy, and then they changed, helping the different states how to manage the pandemics, and then buying special or providing units to produce oxygen or things like that. And then they realized that perhaps they should invest in trying to understand what was going on in Brazil. And they started funding projects for sequencing and to doing some epidemiology of the disease. And then it was at that time that we were really able to understand what was going on in the country. And then in the beginning of last year, they said, well, with the remaining money, what we're going to do is actually we're going to create an institute that will invest in helping the country to get organized for preparedness for new pandemics or new epidemics. This was created last year, and then they asked me to be the president of this institute called Todos pela Saúde, All for Health. And the idea is to fill in the gaps and try to help the different places and different activities that are being done in Brazil in order to be prepared for new pandemics. We don't intend to be the ones that are going to do everything. It's absolutely impossible. But there are many groups that work isolated, and as you know, as a scientist, we always try to get the maximum data that we can and hide these data as much as we can, since we have a very interesting paper to publish. This is very good for science, not very good for public health, because we need to see on time what's going on. So we are actually pushing people, scientists from the country, to work together in networks and make public their data, or at least publicize inside these networks, and try to understand what is going on. And that's what the Institute is doing. And the idea is to evolve to a One Health system where you can do surveillance in different steps. And as I said, again, we're not going to do this together. We are doing by using the FAPESP networks, or the different networks that exist already, but putting these data together. So, and the nucleus of the Institute is just a bunch of bioinformatic guys that have been trained in epidemiology or in virology or in genomics, and they can help different groups, because we realized at the early beginning of the Institute that one of the big problems and the big challenge that we had is how we're going to make all this data that is generated in Brazil in actual information that could help in public health. It could be very important in decisions in public health. That's what we are doing. So this is the Institute, as I mentioned here to you. So it's a non-profit organization created in 2021. And the idea is to collaborate in the development of immunological surveillance system to prepare Brazil to face future outbreaks. So it's really working with other people. And for that, we articulate networks to obtain relevant scientific information to help the country in public health decision making. So let's see, when you look at the number, the evolution of the number of SARS-CoV-2 genomes in Brazil, as I mentioned to you, in the first year, we haven't done anything, although the technology was there. And then in 2021, we started growing the numbers, and this was clear in May, and then the numbers went up. But, so by October, by last month, we had something like 183,000 complete genome sequences that were provided to GISAID, over 34.6 million reported cases in Brazil. But the problem is that still we take 54 days from sample collection, from the deposition of the genome at the GISAID, that's the global bank on these genomes, and that's too long, too much. 2 months is too long. We need to know at the week basis what's going on in the country. BQ1 is here, B5 is here, is not here. We need to be much more agile. And how we could do this, that's another problem that we have to solve. When you look at the other viruses that are important in the country, how many of those sequences have been done and put in the gene bank or in GISAID? So yellow fever, just 195. Dengue, just 281. And Zika, only 76. So the point is, do we know, for instance, dengue that's so prevalent in the country, if the dengue that we have here is still the serotypes 1 to 4 that have been described 10, 20 years ago? I mean, is this important to realize for instance, the vaccine that we're developing here in Brazil for dengue should be adapted or not? Do we have any idea if the different dengues could be related to different clinical presentations of the disease whatsoever? No. And I think we have to do it. Nobody's going to do this for Brazil. So in order to see what could be done in order of sequencing in the country, we try to understand who are doing sequence and how many of these, what technology they were using, and how much sequence they could perform. And those are the three technologies being used: the nanopore, the ion torrent, and the Illumina, by far the most important number of sequencing. And those are the different places in Brazil that could sequence, and the number of sequences they have performed. So most of the places did not perform that much, but there was an effort at least, and we can see that these places could do it. So if by any chances we need, we can provide whatever they need, and that's the role of the Institute. Because the Institute, we can buy reagents, but we also can, we can pay for a nurse or a technician or whoever in order to do or perform the sequencing wherever we need. But what would be important for Brazil is in the future is to have a complete network of people that could perform this sequence wherever it is, and have a sequence system that could be done systematically to understand what's going on. And I don't have the time to tell you how the Institute is approaching all these questions, because we are actually even giving courses to people around the country, just to them to understand how, how important it is to do the genome, how to perform the analysis of the data. And for instance, just for the analysis of the data, we created a course, and in 2 weeks there were 10,000 people that they did registration and are actually performing the course. So, that means also, there is a need. Every time you offer something, there are people jumping. So I think that we know that due to the efforts of FAPESP for sure, but by Esther Sabino, in 2 days we had the sequence of our virus here in Brazil. And this was done because there was already a platform that was established. And Esther Sabino was the one that was responsible in our Tropical Medical Institute to perform those sequences, isolate virus, cultivate virus, and do the sequencing. And this was done in close collaboration with Oxford University, where there are many of our students there as well. And in 2 days they could do. And they did a tremendous effort and tremendous work following the disease, not here in São Paulo, but mainly in Manaus, where the problem was, as you know, the gamma variant started that, and it was a strike. Who else did sequencing? Fiocruz is pretty well organized as well, although they are very close between themselves. Sometimes you don't have access to all the data that you would like to, but they have performed many of the sequences, and they have labs because they have institutes spreading around the country, and they have performed many of those sequences. There is also a network called Virus Network, or Rede Vírus, that performed very well. It was organized at the early beginning. I remember we went for the first meeting in Brasilia in February 2020, when the disease was just starting, and the Minister of Science and Technology created this virus network coordinated by Fernando Spilke from the south of Brazil, and they had labs spread in the country that also collected virus and did a lot of sequencing. And then Butantan also did a lot of work here in São Paulo. Normally, we would expect that Adolfo Lutz would do this job, but Butantan was quicker and got organized and did all this sequencing as well. And also the magazine Luiza put the Women for Science in Brazil together, and also trained and have performed some sequencing, especially in Minas Gerais. So these were the main groups, but I think that we should also refer to the DASA, that is a private laboratory, clinical chemistry laboratory, that performed a lot of sequencing and did a lot of work, and they helped also giving more than 7,000 sequences. In our institute, we said, well, but this is taking too long. What can we do if you want to follow what's going on in Brazil? So what we have done is we had taken data from private labs in the country that were spread in the country, and by, as you know, when you had this Omicron variant, there is we lost a place where for the PCR amplification of the spike. There is a deletion there. So you would not amplify the S. And so with the PCR, you could identify if Omicron was in or not by just looking at the PCR. So that's what we have done by working with labs that use that technology. And we are able to show by the beginning of December, when people were discussing if Omicron would come to Brazil or not, we could identify that Omicron was already in Brazil. And this was the Palmeirense line there, the green line, and that you can saw that it went up, and that was Omicron during the month of December, and then it went down, as you can see there, and then we have the resurgence of the B4 and B5, and so on and so on. And this was followed also, my time is over. And this was also followed, we did another strategy in order to know if the disease was important or no. You remember we didn't have any data from official data. So we took data from the private labs that were performing PCR for diagnosing of people with flu symptoms. And as you can see, very good. So here, this is highly positive, and then Omicron, the B1 is out, it goes down, and then you have here 4 and 5 up again, but you can notice here that kids were not affected anymore, just there, and then it went down again. So we could follow, and this was real time. That means we could say the week, what was the variant was in, number of cases, how important the disease was, and so on. We could also follow, as you can see up there, that other viruses were important, because then we started getting more information from these private labs that were doing syncytial virus, influenza A and B, and we could follow as well and show the population what was going on at what particular age. You can see that syncytial virus, as expected, was more relevant in kids under 4 years old. And we could see 3 weeks ago that the positivity was important again. And we were able to say, now we know that the disease is important again in São Paulo, but we could preview it 3 weeks just before it actually started to be noticed by authorities that disease was coming. So all this, I think it's very important if you want to follow a disease, and you have to get organized. And all this can be done if you get organized and you can pull data. So, just in conclusion, sorry if I took 2 minutes more, Brazil has dramatically expanded capacity to perform SARS-CoV-2 full-length genome sequencing across the country through state and private funding initiatives. Actually, the capacity was there. It's just a problem of investing. Genomic diversity of other epidemic viruses remains poorly understood, as I showed to you. We were taking 54 days to data collection to submission, and that's terrible. And this is the way that we scientists work. It's still a long way to go between sample collection sequence and information to the public for SARS-CoV-2, but even longer for other viruses. It organizes several initiatives to improve cost effectiveness and sustainability of target and non-target metagenomic virus sequencing. And that's what the Institute wants to help, and in talking, of course, other institutes to work together and provide this kind of service for the country. I'd like to thank your attention, but before, I'd like to thank Esther Sabino for putting the slides and being here, and also Anderson Brito and Marco Bergatti. Thank you very much.
Okay, so our last speaker before coffee break. We are moving from viruses to parasites. I would like to invite Dr. João Marcelo Pereira Alves. He is a biologist and is currently an assistant professor at the Biomedical Institute at the University of São Paulo. His main contribution to the field of genomics is in the evolution of trypanosomatids, a group of protozoan parasites that are key to some of the most relevant neglected diseases worldwide. Thank you, João. Please take the floor. Oh, questions, I forgot to say, we will also have time to question and answers during coffee break, so please.
Good morning, everyone. It is a pleasure to be here talking about our work in the past few years, especially work that has been sponsored by FAPESP. And it's going to be a mix of many things here because we work on several different genomes from several different groups. So as the title says, I work on protozoa and bacteria. I have a long history of being sponsored by FAPESP, and I'm very grateful for that. I was a master's student and then a PhD student, and then I got a technical training and a Young Researcher Award in those dates there. So it's been a long relationship for which I am very grateful, and I hope it continues for a long time still. I was actually impacted by the Genome Project even before I started working on genomes. I was a PhD student at the time when the Genome Project was going on, and it impacted me with the capabilities that were built in the state. Professor Luiz Nunes, at the time at the University of Mogi das Cruzes, he helped a lot with the sequencing. I wasn't working on genomes, as I mentioned. I was working on the ribosomal small subunit gene for some amoebas that I was studying at the time. And he helped me a lot with the sequencing of those, with the capabilities that they had, the equipment they had, and I learned a lot about sequencing at the time, and it put in me the bug of sequencing, which became what I do until today. So what we do today is what I like to call protozoa genomics, which basically is a made-up word that means we study protozoans and some other organisms as well, as you're going to see. We use high-throughput genomic methods, so mostly genomics and transcriptomics. And of course, we use a lot of bioinformatics. We develop some tools for our own use, or of course, we use the tools that everybody provides in the literature. And the organisms we studied are concentrated in a few areas of the tree, but mainly that blue circle up there, which is the Euglenozoa phylum. That's where trypanosomes and leishmania, the well-known Euglenozoa, are located because they have great, as I'm sure most here know, they have great medical and economical importance in agriculture and especially in animal husbandry. We also work on some apicomplexa many times with Plasmodium and sometimes even on animals. I collaborate with people from Butantan on the analysis of genomes from mosquitoes and other animals like that. But we concentrate on the kinetoplastids. And I mentioned bacteria in the title of the talk because we also work a lot on beta proteobacteria up there, but specifically beta proteobacteria that are endosymbionts of trypanosomatids. They live inside. They're almost organelles. They evolved, they co-speciated and co-evolved with the trypanosomatids, and they are inherited vertically. If you remove the bacterium from the host, the bacterium dies. So it's almost an organelle, I like to say, although it doesn't fit all the criteria for that. And also we work a lot, as we're going to see quickly, with Clostridium, especially Clostridium scindens, which lives in our intestines and makes secondary bile acids. I work on that in collaboration with colleagues from Illinois, who do the biochemistry part, and we do the genomics part and evolution part here. So we are an in silico lab, important to mention as well. So here are the euglenozoa, for those of you who are not familiar with how they look. We have the famous euglena, the green one. Usually they are free-living, and it's very well known as one of the first organisms to be seen in a microscope. Then there's, that's a Trypanosoma brucei from the blood, representing the kinetoplastids. And there's Diplonema, which is neglected of the neglected, so to speak. They are marine organisms that don't do anything important medically or economically, but they are ecologically important, very important. We learned recently, not us, but the literature. Until a few years ago, we knew only 6 species from 2 genera, and then metagenomics of seawaters shown that they are the most, probably the most abundant and most diverse eukaryotic organism in the seas. So that's the importance of this new technology that we should invest on, looking at hidden diversity that is out there. So going to the trypanosomatids, which is the focus of our work in our lab, it's contractually obligated to mention the tri-trip, right? 2005, the first 3 genomes of trypanosomatids came out, Trypanosoma cruzi, brucei, and Leishmania major. There were Brazilians involved here, and they worked on the Xylella project, so it's part of the, some of them at least, I think, worked on that, as far as I remember. Can't find the names here, but it is a big project with parasites that did benefit from the capabilities built by the FAPESP project. And over the years, we started with those 3 in 2005, and here I collected the dates of the genomes being published on the NCBI website, on GeneBank. This is not perfect because when the genomes get updated, the date changes. So this is not exactly the date of the first sequencing of the genomes, but you can see a curve that goes higher with time there. But there's a small problem for us, at least, who study the more basic, so to speak, the not medically important trypanosomatids, which is that if you get the abundance of the genera that are being studied and make a sort of word cloud kind of graph, you see this. It is mostly, of course, the ones that are medically and economically important, Leishmania and Trypanosoma. So in my project, that was the Young Researcher Award that I got in 2013 from FAPESP, the idea was to study a broader diversity of trypanosomatids, looking at genera that infect only insects or that are closely related to Leishmania and Trypanosomes, to further explore the diversity in the family and actually the phylum Euglenozoa as a whole. So we can see here some of the small ones in black have 1 organism sequence so far. That is published, of course. And Leishmania has 66, so it's a big difference. But we're catching up with time. First, I'm going to talk about some results on the endosymbiont side, which is a very interesting story that we collaborate a lot with Maria Cristina Mota from Federal University of Rio de Janeiro. And we also collaborated a lot with Ana Teresa Vasconcelos at the time, where we studied this relationship between the trypanosomatid host, the eukaryotic cells, and its bacterium. It's 1 bacterium per cell in that computer-reconstructed image that you see there, made by Maria Cristina Mota in a 2010 publication. You see the thing that looks like a green peanut, that's the bacterium. It stays close to the nucleus, and it's just 1 per cell. They divide together. It's quite interesting relationship. And we looked at their complete genomes, the genomes of the host organisms and the genomes of the bacteria. And here first we looked at the structure of the genomes of the bacterium. We saw that they're completely synthetic, which means their genes are all in the same order, in the same positions, even after about 100 million years that they separated. That's, for people who study bacteria, is not something you see very often, unless you're studying endosymbionts. Then you see that kind of phenomenon, and we saw that in this group as well. And mainly, we saw the metabolic collaboration between the host and the bacterium, where the bacterium was, quote, enslaved, was kept inside to provide important metabolites for the trypanosome, trypanosomatids, not trypanosome, These are Angomonas, Trigomonas, and a newer genus, Centomonas, that inhabit the guts of Abertura in general, flies, mosquitoes, things like that. And we can see here, TPE is the acronym we used for the bacterium. It produces several amino acids that the host can use. And RT on the other side is a regular trypanosomatid, as we called the trypanosomatids that don't have bacterium inside, like Trypanosoma, Leishmania, Herpetomonas, things like that. And they have to import from their hosts, the host they're parasitizing, and then they can interconvert. And also take notice that some things have a dark gray border, and that's going to be important in a moment. Just remember they're there. We're going to see that dark border here as well. Here is some, I'm not going to get into detail, of course, of these pathways. These are pathway synthesis for important amino acids, the essential ones, the ones that the regular trypanosomatids have to get from the diet, and we as well. We can see that sometimes pathways are partly made by the bacterium. The bacterium color is that one that says TPE up there. Some are present in the genome of the regular trypanosomatids, some are present in both. Sometimes we have just the bacterium making the complete pathway, as you can see down here for histidine. It's all in the bacterium color. Also up there, shikmate pathway. So we've shown something that was known biochemically for many decades. We pinpointed the genetic basis for all the phenomena that had been seen before. And not just with amino acids, we did that as well with other important metabolites like the heme compound, which we're going to talk a little more in a moment. And this was all done in close collaboration of Professors Marta Teixeira and Ernest Camargo here from USP, which are still close collaborators of mine. We still work together on many genomes. And as you're going to see, sometimes the genome is not a focus of the work, but the importance of the capabilities and the funding for the genome sequencing helps other projects that deal with other aspects of the parasite biology. Now, the gray boxes, the gray highlights that I mentioned, that was another interesting discovery we made when we looked at the amino acid pathways, which was that almost half of them seem to be of bacterial origin, but not from the endosymbiont. So doing the large-scale phylogeny of all of the genes that were involved actually in those pathways, we saw that about half of them came from bacteria, and some of them were ancient transfers. They were transfers that were present both in trypanosomes and leishmanias and herpetomonas in the whole family. So they were not due to the bacterium endosymbiont. They were previous. So that's another level of collaboration between different organisms that we can discover when we looked at whole genomes like this. In the bacterium, we didn't see anything. The bacterium didn't get any new genes. There's an example here. Homoserine dehydrogenase is the enzyme in this case. The one in the bacterium, it's there in the family where it should be. In the beta-proteobacteria, alkaligenaceae family, no problem. In the trypanosomatid, the gene is amongst the Firmicutes, which is a bacterium like Clostridium, Lactobacillus, Streptococcus, things like that. So very far from other eukaryotes. So that's one kind of work we do. Another thing that we solved with the full genome sequence was an ambiguity that we had in these organisms, taxonomy. These are the genera that contain the bacterium inside on the left. So Strigomonas, Angomonas, and Centomonas, which was discovered only in 2014, so after our work. And on the right, you have the bacteria. And as I said, they co-speciate. So whenever the trypanosomatid speciates, and you have two different species of trypanosomatid, the bacteria also speciate. You can see that in the tree. The trees correspond to each other. They mirror each other, except for where I put there a little caution sign. There was an ambiguity in one of them, which was discovered when the Angomonas ambiguous organism was first isolated by Marta Teixeira in 2011. They published it. And they saw that the endosymbiont for those two species, ambiguous and DNA, was the same. But when you looked at the nuclear genome, the ambiguous genome was closer to the sooci. So it looked like there was a switch, a host switch, so to speak. It's not the bacterium is not a parasite, but let's say a host in this case, where the bacterium from DNA got to ambiguous. So to solve that, at first it was done just with 2 genes, so we wonder if it was an artifact. Because when you use few genes, you can have population genetics effects that can trick you in the phylogeny, and that happened more than once in the history of trypanosomatids. So we had the full genome sequenced, and recently, a couple of years ago, we published a phylogenomic analysis with thousands of genes in the case of the trypanosomatid and the full genome pretty much of the bacterium to show that the ambiguity is real. And there was switch, and probably more than once, where they have, the bacteria have been replaced in the lineage there. So we can see that pattern in the tree. The blue part is the ambiguous hosts, and the red part on the side is the bacterium host. We also saw an interesting phenomenon, which was that Centomonas orthogonicus, another trypanosomatid that has bacteria inside, has lost the synthesis of heme, which was always considered as somewhat of a hallmark of these organisms. The classical test used to know if the trypanosomatid had an endosymbiont inside was to put in a culture medium without hemin, which is a source of heme. If the trypanosomatid survived, it means it has an endosymbiont making heme for it. If it didn't survive, then it didn't, obviously. If you kill the endosymbiont, which you can do with antibiotics, then you need to put heme in the medium. That's pretty obvious. Problem is that when my student, he was an undergrad student, and I said, oh, get the heme pathway, characterize that to get used to the methods, because it's always there, it's an easy target, so you get acquainted with this. And he came to me and said, I found only 2 genes. First thing you think, the student did something wrong, right? So we go there and look, and no, he was correct. He ran all the programs correctly, and he interpreted everything nicely, and this organism lost it, so we cannot rely on the hemin test anymore to know if there is an endosymbiont there. And that was quite interesting because we thought it might be another organism that does not survive, that can survive, without heme, but it's not the case. Phytomonas, which infects plants, it is a trypanosomatid that lives in plants, is the only organism we know that can live without heme in a certain condition in the host. But unfortunately, it was not the case. A PhD student of mine, during his master's, he also characterized gene families using all the genome analysis, genome sequences we had available. That's another kind of work that we do a lot, and Marta Teixeira's lab does a lot as well, which is to get all these genomes and focus on certain families. So he was looking at endotrypanon more specifically, and he found, for example, that some surface protein families are greatly expanded in endotrypanon. We have some hypotheses for why that happens. We're not sure, but that gives us an idea for experiments that we're talking to other people in the department to try to test to see if that helps, because it's a parasite that lives in blood, so it maybe needs more glycoproteins in the surface to survive. We don't know. Another work that I was happy to learn on Thursday night and quickly sent a message to my student. He got a FAPESP stipend approved on Thursday night. He's working on immunoinformatics, which is trying to get as much information as he can bioinformatically using different algorithms and comparative analysis to see which molecules in the parasite are likely to elicit an immune response. So they could be good targets for vaccine candidates, for example. So this is also called reverse vaccinology. We've been calling it immunoinformatics because there's a little more to it than just vaccines. It can be used also for other approaches like drug discovery or things like that. So he already found some interesting candidates that are seen in the literature, and his methodology is working well. So we're hopeful that will go well, David's work. And to finish, just quickly, the bacteria side of things, besides the endosymbiont, is the work we're doing on 7-alpha dehydroxylating bacteria that live in everybody's guts, pretty much. They finish the biosynthesis of bile acids for us. And they have importance in liver cancer, as we see there. We can have liver cirrhosis if these bacteria are in dysbiosis. We have unbalance in these bacteria because they produce androgens. They can have impact in prostate cancer, maybe. So they have several interesting things that we're seeing in their genomes. And my master's student, Kelly, she is studying the pangenome. So we got 17 so far genomes from this species, Clostridium scindens. And the pan-genome is almost closed. If you've seen a saturation curve, it's getting close to flat up there. We have to sequence more, which is the first information we got. And the second, more interesting one, is that there might be 2 species there that we didn't know, because their gene content is quite different. There's 2 blocks that you can see very clearly, which we did not expect, where there's a core genome, which is the part that's shared by everybody. And then you can see blocks. Of course, there's specific things far to the right of the graph, but closer to the left here, we can see big blocks that are not shared between the 2 groups. So that's what we're investigating now. What are these 2 groups? What are the genes in these groups? And do they characterize a new species or not? Which is always a big fight in biology overall. Splitters versus lumpers. You all heard about that. So we gotta continue working on all these things. There's many genomes that we have not colonized. That's the problem with genomics. The more you sequence, the more people you need, and we don't always have, so, and don't have time enough to do all the stuff, but we carry on. So I'd like to thank all the collaborators that were involved in these works. Not everybody we work with nowadays, but the people from University of Illinois at Urbana-Champaign, USP and Virginia Commonwealth University, where I got my postdoc, where I learned most of the genomics I do. University of Ostrava and South Bohemia, and Federal Rio de Janeiro and the LNCC in Petrópolis. And thank you all for your attention.
Okay. Okay, so we'll start the last talk of this morning. It will be performed remotely. It's Dr. Anna Childers. She was unable to join us due to the new wave of COVID. So she sent her presentation to us. It's about 35 minutes presentation. Anna Childers, she is a young computational biologist. She is based at the Beltsville Agricultural Research Center from USDA, where she is the director of the Bee Research Laboratory. She is recognized for her scientific contributions to the Ag 100 pests. So within the pathogen genomics, the idea to present to you the initiative of Ag 100 pests is to make you aware of those bold initiatives to sequence one tick, which is something like this small, very small, pretty much almost single, not, I won't say that, anyway. And this is an ARS initiative to sequence the genome of the top 100 agricultural pests in the US. Her work also led to the ARS and Ag 100 pests being recognized at the forefront of the insect genomics, international insect genomics. So we will hear Anna Childs now. After her presentation is time, we all have time for lunch, and we need to be back here by maybe 1:55, so we can start on time at 2 o'clock. Would that be okay? Okay. So thank you so much. Let's hear Anna Childers. Thank you so much, Anna, for sending your presentation.
Hello, I'm Anna Childers, a computational biologist in the Bee Research Laboratory of the United States Department of Agriculture, or USDA, USDA's Agricultural Research Service. A huge thank you to Marianne Van Slice and the other organizers for inviting me to give this talk today. I wish I could be there with you, but unfortunately I'm gearing up for our holiday season here in the States. I'm excited to have this opportunity to talk to you today about our agency's Ag 100 Pest Initiative and the Genome 100 project, two projects producing high-quality genome assemblies at a large scale. I co-lead the Ag 100 Pest Genome Sequencing Initiative with Kevin Hackett and Brian Scheffler. Kevin is currently a national program leader at the USDA, as well as a lead of the I5K project. I asked him for some perspective on our history with Brazil's genomics efforts, and he explained that those ties began not long after Brazil's 2000 sequencing of the Xylella fastidiosa genome, the causative agent of Pierce's disease of grapevines. It was almost fate, since Kevin Hackett was a collaborator with Dr. Jose Bonvay at INRA, working on spiroplasmas. Dr. Beauvais, shown here, was already heavily involved with citrus greening and had described Candidus xylella species of the bacteria causing greening diseases. Dr. Monique Garnier at the same lab had cultured the organism from a tree in Brazil. Another tie was through Sandy Purcell at Berkeley, who was studying the disease in California. Kevin was collaborating with Sandy on cultivating phytoplasmas. All were strongly connected with Brazil. So it was natural when our USDA agency decided to obtain the genomes of strains of the bacterium found in California that we would turn to Brazil. This effort was led on the U.S. side by the lab of Ed Civarello in California, working with Dr. Jose Fernando Perez, a physicist and then scientific director at FAP-USP. Interestingly, Dr. Marianne Van Slice was a key contact. She was an author on the original Nature paper shown here, and I mention her because she is a member of the Earth Biogenome Project, the effort to sequence the genomes of all eukaryotes. In a way, one can draw a line from that early Xylella genome process, that genome success in Brazil, to the Earth Biogenome Project that ambitiously intends to sequence every plant, animal, fungus, and other eukaryote in the world. To quote the historian Tom Friedman, "The world is flat." And it is certainly flat with regards to scientific exchange of information and cooperation, such as between our countries. We are all the better for it. The sequencing projects I'll discuss today both belong to a couple larger umbrella projects. One is the I5K, an international initiative to sequence the genomes of 5,000 arthropod species. The I5K predates the larger Earth Biogenome Project, which is really a community of communities like the I5K that seeks to sequence the genomes of all eukaryotes. I regret that I'm not able to list everyone here, but Brazil has numerous scientists and leaders contributing to the project that have been very impactful partners, not only because of Brazil's immense biodiversity, but because of the important insights and contributions they have made to efforts to ensure the project is conducted with the utmost respect for all nations and indigenous peoples, as well as justice, equity, diversity, and inclusion in everything from data sharing to fostering new scientists. In the fall of 2018, the Ag 100 Pest Initiative was undertaken by a group of us at USDA's Agricultural Research Service with the goal of generating reference quality genome assemblies and annotations for the 100 top agricultural pest arthropod genomes as a contribution to the international I5K effort and the Earth Biogenome Project. Thanks to the success of the project thus far, and the great need for genomic resources for pestiferous arthropods, Ag 100 Pest plans have now expanded to over 170 projects. Why do we care about sequencing genomes, though? Arthropods truly have major impacts on agriculture, including animals, crops, and system health, safety, and productivity. Our vision is to establish the critical genomic infrastructure necessary for molecular research that will aid proper identification and taxonomy of species, understanding of their physiology, and the development of targeted pest management solutions. Our success is due in large part to a dedicated team with leaders distributed from coast to coast across the USDA Agricultural Research Service. With teams dedicated to everything from technology transfer and extraction all the way to genome analysis and post-assembly support. We really are taking specimens from extraction all the way through public data dissemination and functional gene annotation. I truly can't thank this team enough for their dedication to this project. When it came to choosing the quote-unquote top 100 ag arthropod pests, we consulted with several groups that work on invasive and agricultural pest issues, as well as USDA researchers and the broader arthropod research community. We knew we needed to address diversity across agricultural stakeholder groups, including field crops, animals, bees, forests, and stored products. Additionally, while our focus is on U.S. ag pests, we also wanted to keep certain pests across the world in mind because of their high potential to become established as invasive species. In the end, we chose species most requested by the community members to advance their research programs, and we've expanded our efforts through several collaborations. When we started, we embraced the Earth Biogenome Project's goal to generate high-quality genome assemblies. By doing so, we hoped to generate reference assemblies that would stand the test of time, rather than being drafts serving as part of an iterative effort. On the x-axis, this plot shows the quality of arthropod genomes that had been generated by October of 2018 when we began the Ag 100 Pest project, and the number of genomes is on the y-axis. Please note that a log 10 scale is used on the x-axis of this plot, so a 6 represents a 1 megabase pair N50. As you move along the x-axis, you can see whether those assemblies were sequenced at a poor level on the left side or a high level, the arrow moving to the right side. Poor means there are millions of small DNA pieces in the assembly. Good means just hundreds of very large pieces of DNA, where we can not only find genes, but also important regulatory regions. In practical terms, the poor assemblies meant lots of holes where we don't know the true DNA sequence. You can see from the blue box that only a few of the arthropod genomes in the NCBI genome repository were at the level that we wanted, and that meant the EBP standards at the time. Our goal was to produce genome assemblies that were on the far right of this plot, but most genomes at the time were falling far below the quality we were aiming for. A few projects were generating high-quality assemblies, but no projects were yet producing high-quality arthropod genomes at the scale we planned. Our goals were certainly audacious. Not only did we want to produce genomes at scale, but we also wanted to produce them from a single specimen. In 2018, the assembly metric goals were only a scaffold contiguity of 1 megabase pair N50. But with technology improvements over the years, including the release of PacBio's hi-fi sequencing technology and ultra-low input methods, the sequencing targets have also increased to where we are now, with the goal of having the contig contiguity rather than the scaffold contiguity at least 1 megabase pair N50. And the scaffolding now, we want that to be at the chromosome level. The most satisfying part of the Ag 100 Pest Initiative scientifically is to see that we have been able to obtain genome assembly qualities way out on the right side of our target area, well within the most recent, more stringent Earth Biogenome Project assembly metric targets. Technically, we are where we need to be. This is important to enabling arthropod genome sequencing more generally. We had our goals, but we needed a proof of concept, so we partnered with PacBio to use their new technology as it was being released. Single specimen sequencing is particularly difficult when you're trying to sequence a tiny insect because you can only extract tiny amounts of DNA. This is especially a problem for small insects that have large genomes because, as you can imagine, the larger the genome, the more DNA you need to sequence in order to get sufficient sequencing coverage. This is why we were ecstatic when our scientists succeeded in sequencing the small spotted lanternfly insect, a new invasive species, one with a large genome, over 2 gigs. In fact, it was the first insect genome published using only a single SMRT cell on the Sequel II sequencing platform. This partnership and success essentially made possible our sequencing goals. Although we started the project using PacBio's long-read CLR or continuous long-read sequencing approach, we have been quick to adopt improvements to the technology. We now use hi-fi reads, also known as CCS or circular consensus reads. Because these have shorter subreads, lower nanogram amounts of input library are needed for loading, allowing us to target smaller species. Hi-fi reads also have a far lower error rate, such that we can produce a highly correct and contiguous assembly with lower sequencing coverage and no Illumina polishing. Another bonus is that assemblies generated from hi-fi reads take less compute time, which is important when you're generating genomes at scale. This is a depiction of our genome assembly pipeline utilizing PacBio hi-fi reads and Hi-C scaffolding. As we continue to embrace technology and software improvements, our genome assembly pipeline continues to evolve to increase the quality of the assemblies we are producing. Over the last several years, our embrace of progress has allowed us to tackle genome projects for arthropods of increasingly smaller physical body size and larger genome sizes, all while increasing our speed of sequencing and the quality of the data and assemblies we are producing. That progress was put to the test in 2020 when it became clear the Asian giant hornet, now renamed as the northern giant hornet, had been introduced to the Pacific Northwest. Not only did we sequence it, but we set a new bar for speed from specimen capture to final sequence release, just weeks compared to years, which is more typical. It therefore has set a new paradigm we hope will be employed on other invasive species challenges in the future. I mentioned we have also been able to sequence increasingly smaller specimens. Over the course of 2020 and 2021, it was our improvements in the extraction and preparation of those extracts for sequencing libraries that has enabled us to sequence far smaller species from a single specimen than was possible when we started the project. In fact, many of the species we work on are very small, so much that our standard approach is actually a low input approach for high fidelity or hi-fi PacBio sequencing. Here I am showing examples of several grain pests. As I mentioned, the EBP, or Earth Biogenome Project standard, is a 1 megabase pair contig N50 for specimens with sufficient DNA. As you can see, even with species that don't actually have ample DNA to spare, we are exceeding that mark. For especially tiny species, our team was given the opportunity to beta test PacBio's latest ultra-low input technology. This amplification-based technology allows us to still utilize our single specimen sequencing approach for arthropods far smaller than we dreamed possible when the project began 2 years ago. Despite the size of these species, the assemblies we have generated exceed the high metrics that have been set for species for which the quantity of DNA is not limiting. This has been a great advancement for arthropod genomics, and in the long run helped PacBio eventually market the kit to arthropod sequencing groups. The species I'm showing here are all grain pests, including Tribolium castaneum, otherwise known as the flower beetle. Tribolium castaneum was actually one of the first insects sequenced and is a model organism for laboratory study. Here you can see an alignment of our Tribolium genome assembly on the bottom against the current reference assembly at the top, with the chromosomes represented by the gray blocks. The spaghetti mess of colored lines between them is a mapping of known genes. Not only has our more contiguous assembly improved the proper position of genes in the genome, it is also about 1/3 larger than the current reference assembly. The arrows here indicate some of this new sequence. For a long time, researchers have known biologically that this extra sequence exists, but no assemblies have ever been able to capture it. Now, researchers can work with confidence as they compare this assembly to others. After all, you can't say a gene or a region is missing if your assembly isn't complete. Here, our Tribolium castaneum assembly is now at the top, and another Tribolium species we have sequenced is below. Now you can see that through evolution, genes have moved around on the chromosomes, perhaps freeing them from genomic constraints and allowing the species to exploit different environments. This sort of analysis was much harder with lower quality assemblies that didn't have a full representation of the genome with properly placed pieces. Truly, before you can do comparative genomics, you have to do good genomics first. This is also a great example of our efforts to move beyond single assemblies to comparative genomics, in this case, with some stored product pest insects. In addition to sequencing 8 Tribolium species, we are sequencing numerous stored grain pests, including all the ones shown here in blue, plus others. One of the main purposes for sequencing such a diversity of insects across a range of different families is to see whether there are convergent adaptations in these diverse lineages of stored product insects that provide them with the ability to sense and respond to odors associated with stored grains. By unveiling common mechanisms, we can develop targeted control methods across these problematic pest species, as well as develop tools to distinguish them easily, since that can be difficult to do at our ports of entry when some of these are quarantine pests and others are not. We're excited to finish this group of species and support our scientists as they explore these genomes. I've shown you examples of how we were able to increase our speed of sequencing and the quality of the data and assemblies we are producing. I also showed you how we have now tackled species with even tiny physical body sizes. Well, as of this year, I can now say we've also sequenced some impressively large genomes. This year, one of our big successes was the release of a big genome, a chromosome-level assembly of the almost 9 gig desert locust genome. We were able to resolve all 12 chromosomes, despite some of those individual chromosomes being over a gig each, which is multiple times larger than many entire insect genomes. But actually, we didn't just release one locust genome. Together with the Behavioral Plasticity Research Institute, we actually released 6 locust genomes. This was an incredible effort that produced assemblies of unprecedented quality. As you can see by comparing the incredible contiguity of their contigs and scaffolds to other insect and locust assemblies. As a first pass at comparing the 6 locust genomes, this Centenary map was created using 1,300 single copy orthologs, which were all annotated ab initio. The species were ordered based on a phylogeny constructed from about 1,200 genes. Looking at all the genomes at once enables us to identify regions and patterns to further investigate. As you can see, among these beautiful genomes, there are quite a few inversions, which are some of the first places we hope to start looking. We also annotated repeats in these genomes and provided the models to NCBI to assist in gene annotation. We also generated pie charts of the fractions of the genomes attributed to various classes of repeats. The black region of the pie charts is the non-repetitive portion. We found that each of the genomes examined so far contains around 70% repetitive content. These repeats contribute to the large size of Schistocerca locust genomes and to challenges in genome assembly. It's valuable to classify transposable elements as families because they propagate differently and can have different impacts on recombination, methylation, and gene expression, for example. We use these data to generate interspersed repeat landscapes that help us infer how repeats have propagated through evolutionary time by estimating divergence rates between the classified repeats. Across the locusts, we can see that along the x-axis, the age and timing of repetitive bursts varies between species. If you're interested in learning more about the grant-funded Behavioral Plasticity Research Institute, or phenotypic plasticity in general, and how these 6 locust genomes are helping us decipher the molecular mechanisms behind it, I would encourage you to read this blog post on the DNA Zoo website that we published with the release of these 6 genomes. Now, after we finalize genome assemblies, we submit them to NCBI, where you can monitor our Ag 100 Pest umbrella project, shown at the top here, to see the latest releases. Once the assemblies are publicly released and gene predictions have been performed by NCBI, the I5K workspace team at the USDA National Agricultural Library runs functional gene annotation and provides support for the genomes with numerous visualization and search tools. They also provide support for manual gene annotation. The work they perform is really a unique resource not provided by other genome sequencing initiatives. In addition to all the species I've highlighted today, we've been working on many other assemblies too. These pie charts give you a sense of the diversity of species we are working on. We have now sequenced over 130 specimens across 8 orders and 43 families. Through the Ag 100 Pest Initiative, we are helping to greatly expand not just the number of arthropods with reference quality genomes, but also the number of arthropod families with a reference genome, thus filling important existing gaps. As you can see by comparing the number of families per order for which we have produced data in the pie chart on the left to the number of assemblies per order in the pie chart to the right, we have in some cases produced multiple genomes per family. As I showed you with the stored grain pests and locusts, we are looking beyond genome sequencing and toward comparative genomics. You can learn more about the Ag 100 Pest Project on our website here, as well as through the Genomes on a Tree portal, also known as GOAT, which is produced by the Wellcome Trust Project, where you can also see what genomes are being produced by other Earth Biogenome affiliated projects. Now, I want to transition to pollinators, which are critical insects for natural and agricultural systems. As you know, bees serve as vital pollinators and are very diverse. New tools are needed to facilitate bee identification and monitoring and to provide insights into pollinator biology across species. Bees are also under threat from multiple factors, including pesticides, pathogens, and climate change. Genome resources are needed to identify risks to species and to mitigate negative effects to species and populations. Many bee species are listed under the Endangered Species Act or are being considered for listing. Genomic resources are needed to study the genetic diversity of populations and to help conserve species. By building on the success of the Ag 100 Pest, when the USDA National Pollinator Center in Mississippi was founded with a focus on pollinator health, honeybee sequencing was part of that mission. One project they undertook using our pipeline was the sequencing of the important honeybee breeding lines with a vision of a honeybee pangenome. Here I'm showing the first of those assemblies compared to the current reference honeybee genome that is available in NCBI. The arrows indicate whether the ideal number should be larger or smaller. Across the board, our assembly is superior. Now, I don't know if you'll be impressed by how much of an improvement our assembly is, but just consider that our assembly used less than 10% of the input resources that the current reference did, and was the product of a few scientists rather than an international multi-year effort. Certainly, the sequencing of genomes has changed. One thing I find especially amazing is that we are now at a point where these statistics are becoming meaningless because we are able to sequence through an entire chromosome in a single contig or contiguous piece of sequence. From this high-C contact plot, the blue boxes are chromosomes and the green boxes inside them are the contigs. For this assembly, we were able to assemble 4 of the chromosomes as single contigs. It still amazes me that we can do this at all, but in fact, it's becoming the norm, and we are now getting to where many or most chromosomes in an assembly are coming together in one piece like this. So what's next? We are working to sequence the remaining bee breeding lines to generate a honeybee breeding line pangenome, which will inform the work of researchers searching for better ways to breed a better bee. Continuing to build from that bee work, and thanks to some congressional funding and a partnership with the University of Illinois, we now have the Genome 100 project. This project has quickly gathered steam. from an early focus on collections to amazing progress on sequencing. The genomes coming from this effort will help resolve phylogenomic questions, expand informative markers for population analysis, improve identification, and aid research in multiple fields, including understanding differences in environmental sensitivity and immunity. Species are being selected based on taxonomic diversity, biological importance, and conservation status. Due to amazing collection efforts, this project will cover a wide taxonomic diversity by sequencing representatives from all but one bee family. And that is only because that particular bee family only occurs in Australia. Although I will say, I suspect if it's possible, our researchers will find a way to get that one too. The Genome 100 project involves several USDA research locations across the country, as well as many collaborations with university researchers, some of which are listed here. As a highly collaborative group, many research subgroups have formed around not just taxonomic analysis, but also immunity, neurobiology, sociality, toxicology, and chemical stress, host-parasite interactions, microbiomes, and others. The group's early success in prioritization and collection of diverse bee taxa is enabling quick progress in sequencing these high-quality bee genomes. I would encourage you, if you're interested, to find more information at this website or to contact the group if you would like to join. To date, the project has already produced hi-fi sequencing data for over 60 species. In this chart, you can see that not only are the initial contig assemblies highly contiguous based on their position along the y-axis, but what might surprise you is how large some of these assemblies are. That surprised us too. But that's because these initial assemblies haven't been decontaminated yet. And what we're finding is that our sequencing methods and the inherent diversity of organisms in and on bees has presented us with a unique opportunity to not only understand bee genomes for the breadth of bee taxa, but also their metagenomes. For all bees sequenced in this effort, taxonomic identity and metagenomically derived DNA can be extracted both from the raw data as well as assembled contigs for the purpose of determining metagenomic diversity in bee species. We are finding that once these other genomes are removed, the assemblies of the bees themselves are of the expected size. Our microbiome researchers are extremely excited, though, to have these additional assemblies to better understand many aspects of how much these other pathogens and symbionts are shared between species, and to understand the microbes themselves with high-quality assemblies. I hope with this insight into Ag 100 Pest and Genome 100 that you can see that we endeavor to push the boundaries to produce better genome assemblies, regardless of a specimen's physical or genome size, across a wide array of the arthropod tree of life. However, while I may have emphasized the assemblies we've generated through the sequencing and assembly pipelines we've built, we are not just service providers. We are all researchers behind these efforts. It is the biology that drives these efforts, and working with our genome champions to produce resources and perform analyses that enable their work. We're proud of putting these assemblies in the hands of researchers as tools in their efforts to find innovative ways to decrease the impacts of these agricultural pests across a wide array of impacted commodities, including field crops, animals, bees, forests, and stored products, and continue to protect the health and diversity of our pollinator communities. Thank you again for this opportunity.
Hi, we have Anna online, I think, right? Hi, Anna. How are you? Hi. Whoops. Where is she? Ah. We cannot hear you well. No. It's very, very low, your talk. So anyhow, I would like to thank you so much because it was a great presentation, and we are sad we would like to have you here next time. Yes, that would be wonderful. Now we can hear you.
If there's any questions, I would be happy to try to answer them.
And I think we won't have time to have questions right now, but with your email, I'm sure people will contact you because it was very inspiring. Not only the size of the insects and also the beautiful bees, but also the quality of the assembly. Thank you so much, Anna. Thank you.
Thank you, and I have one comment, everyone. Thank you again for having me.
Thank you, Anna. Gente, agora na hora do almoço, eu vou pedir para todo mundo levar todas as coisas, porque o auditório será fechado e a gente não se responsabiliza por qualquer coisa que seja deixado aqui. Vou pedir, por favor, para todo mundo estar de volta a 1h55, para a gente iniciar pontualmente às 2h. E aí eu vou passar o bastão para o Luiz Eduardo Aranha Camargo gerenciar a sessão da tarde.
Well, good afternoon, everyone. Welcome to this evening, afternoon session of the 20+2 event, Genome event, initiative by FAPESP. This afternoon, the topic that we'll cover is agronomical and environmental genomes. But before we begin, I will just give you some background information. As you know, the Genome 20+2 event celebrates the sequencing of the first genome of a phytopathogen, the Xylella fastidiosa. We saw this morning. And this was accomplished by the ONSA, the Organization of the Nucleotide Sequencing and Analysis, an initiative by FAPESP. Hundreds of researchers participated in this initiative, and many of them are watching us online. So for them, we extend our greetings this afternoon. And the event is, that was mentioned in the morning by Marie, but the event is structured around 3 sessions. We had a session this morning on pathogens. This afternoon we start with, as I mentioned, the agronomical environmental session. And continuing to tomorrow at 9 o'clock, we'll have the cancer genome session. Each session comprises 5 presentations, one about historical aspects of the initiative, followed by 3 related to actual projects, and after the break, a final one offering perspectives in the field. Before we begin, let me remind you to visit the exposition during the breaks. You can't miss it because it's right by the food, where the food is, in front of the food. So take your time to go through the posters and see some nice pictures from 2 years ago. We also thank our sponsors, Illumina, Eppendorf, and Locus. And it will be interesting, as my colleague Ovesio mentioned to me in the morning, to see how many of the 3 companies are here and how many of you participate in the genome project, either as a scholar or a student, 1, maybe 2, 3. So this is another contribution of the genome project, right? Capacitating, building capacities for the private initiative. Unfortunately, we won't have time for questions after the presentations, but we can use the break to chat and exchange ideas. right? Well, just to give a brief introduction, a contextual introduction of this session, after completion of the Genome Project, the 33 laboratories of the ONSA were assembled into research groups according to their primary interests, research interests. Among these, we had groups of laboratories that sequenced genomes related to the agriculture and to the environment. And we had, like Professor Perez mentioned in the morning, we sequenced sugarcane ESTs, coffee ESTs, eucalypt ESTs, other strains of Xylella, the human pathogen Leptospira, and the sugarcane pathogen Leishmania xylem. So this is the context of this afternoon related. The presentations are related to this, topic. So with that said, let me introduce you our first speaker, Professor Paulo Arruda. Paulo was one of the coordinators of the Xylella project. Afterwards, he led the sugarcane EST genome project, or SUCEST, and acted as director of Allelics Applied Genomics. He's a full professor at Unicamp and coordinates the research Genomics for Climate Change Research Center in partnership with Embrapa. He's a member of the Brazilian Academy of Science. Please, Paulo, come to the stage.
So thank you very much for the nice, kind words. And thank you very much, Marianne, for the invitation to come to this fantastic, historic meeting. And it's a great pleasure to be here and talk a little bit about what we did during the Genome Project and afterwards, and what we are doing now, next steps. So just as mentioned, the early speakers by this morning, starts the Xylella fastidiosa genome sequence started in 2000 from scratch. No one with experience in really sequencing a complete genome of any organism. And it was very nice because when we invited, as Perry said, we invited André Ruffo to come and, as a kind of scientific advisor. And he, I knew André Ruffo from many years. He's a very fantastic East scientist. But when he came to FAPESP, he asked, who's going to do bioinformatics? And I said, what? He said, bioinformatics. Honestly, it was the first time I heard the word bioinformatics. Mendanis used to come to our lab at CEVMEG to talk with, and at the time of Adilson Leite, was a molecular biologist there, and talk about computational biology. But anyway, and when we are about to finish the sequencing of Xylella, the Sugarcane Consortium, CTC, came to FAPESP and asked Paris why we don't sequence the sugarcane genome, because it's a very important bioenergy crop, and this should be very important for the state of São Paulo, the largest producer of sugarcane in the country. And then Paris gave me a phone call, and said, Paulo, CTC is here, and they want to sequence sugarcane. I said, no. Sugarcane is a very big and complex genome. Let's sequence the expressed genes. At that time, that was called ESTs, expressed sequence tag. And then we started the project. But this Xylella, as told by Paris, we had 34 groups working on sequencing and analysis. And then I said to Paris, there are many other groups around the country that is interested in sugarcane research, in sugarcane biology, including northeast of Brazil. It's important. There are important states there growing sugarcane. Why we don't invite them to participate in the project? And then Paris said, but FAPESP don't give money outside of state. I said, okay, but we can call to the state foundations of those states, and then they can contribute. And I said, we don't need to buy sequencers to them. We have sequencers here. Ask them only to buy a computer for a student to use the computer to look our database and find their most favorite gene. And this is what we did. There were many other laboratories from the south to the north joined the consortium for sugarcane ESTs that we call the SUCCESS sugarcane ESTs. And then the question I asked everyone was, look the SUCCESS database and find your favorite gene. What is a favorite gene? Favorite gene is something that are of interest of particular groups. For example, people trying to understand how sugar can accumulate sugar or resist to pathogens and so and so. In my lab, there was a student that was interested in sugarcane response to drought. This is a sugarcane plantation after a frost that killed the sugarcane plants in São Paulo. And yes, he did a fantastic work, a student in the lab, to find the genes that are involved in sugarcane response to stress. And then finish by the end of the ESTs, sugarcane ESTs, I received again a phone call from Fernando Reinach and said, Paulo, come to São Paulo. I am here with the people from Votorantim group that are interested in investing in biotechnology. And I said, okay. And then I came, and then they said, let's build a startup company on to work with sugarcane, eucalyptus, citrus, and then we start by late 2002. We start Allelix. You can see here we trained about 60 people, and then because we learned how to find genes, how to search database to find genes based on expression and so on. So, and this is one of the genes that we found for eucalyptus. Is a transcription factor that we named Waldorf because it's a transcription factor that helps the cells to make cell wall, the most important trait for eucalyptus. You can see here in some of the transgenic events overexpressing this Waldorf. You almost don't see, the vacuous. It's only cell wall, increasing almost 80% of cellulose. And then this is the patents of in companies we don't publish, we don't make papers, we make patents. And then this is one, of course, there are many other patents attached to this related to cell wall and tissue-specific expression. And in 2009, when I was helping running Allelix, I was part-time at the University of Campinas. And then when I came back to full-time professor, I found a very, let's say, interesting time because no one's like to hear about my experience in a startup company. I don't know why. But then I start, to do many things. And in 2013, I was approached by researcher from Canada that was coordinating a very large program on drug discovery, structural genomics consortium that involved the 10 largest pharmaceutical companies and 5 big universities like Oxford, Toronto, Frankfurt, and North Carolina. And then we founded this Medicinal Chemistry Research Center. This was a partnership between Unicamp and Structural Genomics Consortium, and was funded by FAPESP through the PT program, and then became the ENCT Center and MRP. What we do there? From gene, probe. What's a probe? Probe is a small molecule, high potent, high specific, non-toxic, that can target a protein or a target in a cell related to human disease. So this is one of the, let's say, the most equipped and experienced laboratory in the country when we have genomics, structural biology, cell biology, medicinal chemistry, chemical chemists that do synthesize new molecules based on the crystal structure and so on. And then at the same time, I was approached by the director of, the president of Embrapa, saying that, well, Embrapa is running a research program for decades, but we don't have yet, let's say, a blockbuster in terms of trait for agriculture, like BTRR, etc. And then we create a partnership between Unicamp and Embrapa that we call it a mixed institute from Unicamp and Embrapa. And then we receive funds from FAPESP through the Center for Engineering, the program for Center for Engineering Research, and this is what we are running now. Both of those centers have something very interesting in common, and then we brought to the dean of our university saying, we need to create a kind of genomics hub for health in agriculture. It is very difficult to convince them that health and agriculture have a lot in common. What they have in common? Because they use the same tools to do drug discovery and to develop new traits. They use genomics, biochemistry, protein structure, and proof of concept in cell or in plants. The tools are the same. Why we don't put together those guys so that we can, let's say, take advantage of one expertise and another expertise and put together and have, let's say, a synergism. And this is what I set up at the University of Campinas. The Medicinal Chemistry Research Center is dedicated. We only do projects with companies. We don't do pure academic projects. This is because we learned through the Structural Genomics Consortium with a very big partnership with many different pharmaceutical companies. We learned how to manage projects together with the companies. Nowadays, we have partnership with Ache, Europharma, Promega, HiLab, many others. And we have also funding from the NDI, from Bill and Melinda Gates Foundation. And we have partnership with a bunch of international universities. And this is the lab, and this is the team. We have about 15 to 20 postdocs running this project. So it's a very, very, very interesting. And again, we set up all of this from the scratch. The university gave us a space, empty space, and then we brought money and we construct one of the very important labs for early stage drug discovery. Here, for example, is one of the achievement of the center that was published in Cell Reports. We started from what we call a template compound. It's a potent small molecule, but toxic. This targeting as AAK1, that is a kinase associated with many different diseases. And then using structural biology and medicinal chemistry, we were able to develop a potent, non-toxic molecule that is now being used by many different groups around the world and pharmaceutical companies to develop new drugs. This is why we put together health and agriculture, because the expertise we have in the SECMED, in the Medicinal Center, we use to understand proteins in corn that is associated with drought stress. This is the example of crystal structure developed in collaboration with these two labs for different protein kinases that we know is involved in drought stress response. And what we are interested in is developing small molecule that can inhibit those kinases so that we can improve the drought stress response. Okay, let's talk a little bit more in detail what we are doing with the Genomics for Climate Change Research Center. Well, this is a picture showing that 80 to 90% of all we eat comes directly or indirectly from 5 crops: rice, maize, wheat, soybean, and sugarcane. Why? Because we eat cereals, carbohydrates from cereals, legumes, and fruits, fat from soybean, proteins from milk, meat, eggs, and fish. But all of this needs those crops, those seeds, in order to feed the animals to produce. So we are dependent on agriculture for food production. And there is a dilemma of food security, because most of the water, the fresh water, is used for agriculture. There's 40% of the world employment is in agriculture. 30% of greenhouse gas emissions are related to agriculture, 16% for deforestation. And there is a number that's about 20%. We need to increase 20% in production, reduction of gas emissions, reduction of rural poverty, and so on. So this is the challenge. So how to increase the productivity with reducing the impact of all of these the things that affect climate change. So this is only to show you that what happens with increase in temperature. This is the effect of drought in 2015, 2016. And you see here, when you go to red, you increase the temperature, and you go to pink, decrease in temperature, normal temperature. And you see that this here, that only for corn, we had 18 million tons of loss, corresponding to 11 billion reais. Last year, the drought was even worse, and we should expect a very big impact. But this is not only the problem of climate change. The problem is that we use a lot of chemical fertilizers and chemical pesticides in order to produce all the crops for food production. So at we call GCCRC, that's the Genômica for Climate Change Research Center. In the medicinal chemistry, we go from gene to probe. What's a probe? It's a small molecule that targets a protein. And here we go from gene to trait. That is a gene that have an impact on the disease resistance, yield, and many other things related to trait. So there is, same time, we go from gene discovery using instructional functional genomics, gene editing, GWAS, and bioinformatics, and then we use this information to make DNA constructs that we use to transform plants, and then to do phenotype. And the idea is that we go up to what we call proof of concept in the field, so that we can be sure that what we are doing has an impact. And we have a robust research pipeline, and all of these we have is supported by information technology, intellectual property, and regulatory affairs, and so on. But one thing that is very important also, we have been developing a lot of research on microbiome, taking advantage of the genome sequence that we learned at the beginning with the FAPESP genome program. For example, we are working with, the question is, where are the genes for drought, stress, resistance? Those are plants growing in a place that we call Campos Rupestres, that is located around Minas Gerais, northern state São Paulo, Minas Gerais, where we have, let's say, the worst environment in Brazil, with 4 to 6 months without rain, very dry, very poor soil, no nutrients at all, and one of the biggest hotspots of biodiversity. It's very interesting. There's so many biodiversity, so many plants growing there. And then we asked, how those plants grow in those worst, very strange environment? And very interesting, this is a picture, a late rain season, when we have the end of the rain. You see here, this picture show 2 species growing in this area. And then the late drought season, when you have one of the species completely dry, and the other keeps very green. We call this one as evergreen species, and the other one that is dried is a plant that is a resurrection plant. It plants after the first rain. The leaves that were dry becomes green again. It's not a new branch. It's the same leaf that was dry become green. This is called a resurrection behavior. The idea is, how does the plant have this thing? So the idea here is how to sequence the genome and find in the genome the genes responsible for this behavior. So we sequence Veloz intermedia, that is the resurrection plant, and Veloz intermedia, that is the evergreen plant. And this is very important. We heard the previous presentation on how important it is to sequence complex genome like those plants at chromosomal level. And this is what was achieved in our center. We sequenced the genome of these 2 species at chromosomal level, so that we can do can compare the 2 genomes and try to find the genes that are responsible for resurrection and the genes that are responsible for the behavior of keeping green even in a very dry season. So we do a lot of comparison, and this is how we do this, looking for it, because we have the chromosome, we can have the synteny, and then we can compare stretch of the genome to find the genes, the difference from the different genes involved. And this is what we do. It's a very, let's say, bioinformatic intensive work. And this is done in our center. But we don't do only the sequence of the plant genome, we sequence also the microbes that are associated with those plants. Because we have learned in the last maybe 4 or 5 years that the plant microbiome have a big impact on plant physiology. And this is one of the work that was done at the lab, and very important. To sequence the Xylella genome, we have about 100 people, 34 groups. Only one PhD student sequenced 500 genomes, assembled 500 genomes from reads coming out, shotgun reads coming out from the Illumina. This is to show how the genome sequence progressed in the last 20 years. And why this is important, because we now can look in the individual genome of those bacteria and ask the question, what perhaps are the pathways encoded by the genomes that helps plants to become resistant to drought, and how, and what are the pathways involved in nutrient acquisition? Because remember that this soil, this region is very poor in phosphorus and so on. And this is a paper that is now being published in this is a bioarchive, and now it's accepted for publication in a very important journal. And we show that it's very interesting, including viruses that are present in in some of the bacterial genomes that are responsible for lateral gene transfer, and including characteristics that is very important, like nitrogen fixation. So viruses helps plants to exchange information or sequence our genes that are important for plant characteristics. Oops, sorry. Okay, so we can use genomics, transcriptomics, everything, omics in general. We learn how to play with plant genome, with microbe genome, and how to use this to find genes. This is an example of a gene that was discovered in our research center, and we use this is in a collaboration with Nottingham University, and we discovered a drought stress resistant gene. We put the gene overexpressing in barley, and you'll see here, after 12 days without irrigation, the phenotype of the plants with and without the genes. Well, we also did the microbiome of sugarcane, and this was a fantastic work done by a couple of PhD students, then became postdocs, and then became entrepreneurs. I will show you later. And we were able to we were able to, let's say, map the bacteria and fungi communities in sugarcane, in the soil associated with the roots, in the leaves, in the stalks, and everything. And then we map it. This I don't go in detail. This is published, and then we asked it a very simple question: what are the most abundant bacteria that colonize sugarcane? We call those bacteria robust colonizer. And how we determine what is a robust colonizer? Just count the number of the reads that comes from any individual bacteria in a sample. And then we did what we call synthetic microbial community. We picked some of those robust colonizers, and we grow this bacteria, make a mix, and put in a corn plant. And this is very interesting. And this is true what I will say today. the students involved each other. He put the bacteria, inoculated, and after the plants grow up to maturity, and he said, there's no difference. I didn't see any difference in plant development, nothing. And then he said, I'll leave there. Then later on, I will discard the plants. 3 weeks later, he went into the greenhouse, and 3 weeks without irrigation. And what he saw, the non-inoculated plant just bent, and the inoculated plant is very happy growing. And then he said, we discovered the, the microbes, or the bacterial microbes that makes plants to be drought resistant. So they went in a very sophisticated experiments, and they discovered that the microbes help plants to keep the temperature low than non-inoculated plants. And this is the research team of the GCCRC. We have a lot of collaboration with different MBRAP units. We have a lot of collaboration, international collaboration with Dirkinze in Belgium, Hans Lamber in Australia, Juan Imperial at UPM, Rand Wiesler at UD, and Joe Wilson at Nottingham, Maurice Moloney, and many different collaborators in Unicamp and Embrapa. And it's a part of the team. This is a lab where we do genome editing, and at my side, you see Antonio, who did this assembling of 500 bacterial genome using shotgun Illumina sequencing. And what's next? Okay, so I was invited by Fund of Investment, Vesper Ventures. They are a kind of startup builder, and they are interested in very disruptive, risky ideas in biotechnology, in health and agriculture. And I am helping them build up startups in agriculture, plant biotechnology. The first company was Symbiomics. It was created by the students from my lab that, okay, they learned everything they did, they published, but they learned a lot on how to play with the plant microbiome. And then they built this company. You can go to their websites, and they are now being invested on by international funds, and they are growing very fantastic with partnership with big multinational companies. And then I said to them, we need to start a genome editing company, because this is going to be let's say the future of agriculture, the future of sustainable food production. And then we started this company. I invited some of the student postdocs of the labs are interested in joining this with a very disruptive and risky idea of building a new concept of genome editing for trait development. This is what I'm doing now. One of the things that I am doing now. So helping building plant biotechnology in startups, people with high value proposal, and with very risky idea. This is so. Thank you very much. This is my company email, in a bio. company. So if you are interested, go to our website and see what we are doing. We are interested in the genome editing plant, but in this country. Thank you very much.
Thank you, Paulo, for the nice presentation. Interesting. Let's go to the next speaker, Dr. Ana Teresa Ribeiro Vasconcelos. It's a pleasure to have you here with us. Ana is a biologist, and she coordinated the Brazilian Genome Project. Among others, sequenced the Chromobacterium violation genome. You remember that? She's head of the bioinformatics group of the National Laboratory of Scientific Computing of the Ministry of Science, Technology, and Innovation, and Communication, I guess, and member of the Brazilian Academy of Science. Ana is currently coordinating the Rio de Janeiro COVID-19 Omics Network.
Wow. Thank you, Maria, Ana Maria, Fernando, Pacheco, everyone to invite me to be here today to present a little bit outside São Paulo, outside FAPESP. It's a challenge for me to represent the rest of the, the Brazil Bioinformatics and Genomics Network that as I will try to show that start here in FAPESP. And I think that's the reason that I'm here, to say thank you for FAPESP to initiate this program, to coordinate the projects, and to collaborate with other partners around the country. So I'm working. Yes. I will do another thing, Aranha, because I will try to talk a little bit for the importance of the omics and the bioinformatics and the strategic vision that FAPESP had in the past, that, and the challenge that we have in the future. So I'm working in the Institute for, National Institute for Scientific Computation. It's one of the institutes of the Ministry of Science and Technology, and the Bioinformatics Institute is in Petrópolis. My history starts, I will talk to my lab only in 3 slides. And I start with the bioinformatics for this person, this professor, Darcy Fontoura de Almeida, that was a professor in Rio de Janeiro, was my mentor from my master and my PhD mentor. And one day he came to LNCC, my institute, with this book, Sequence Analysis in Molecular Biology. It's the first book that starts to talk about bioinformatics, one of the first books that start to talk about bioinformatics. And in the same time that Antoine Donchamp started with the bioinformatics is the research in silico. At this time, Darcy submitted a project to FINEP, and we had the pleasure to receive Meidanes at the UFRJ, the underground of the UFRJ, to evaluate one of the, I think that's the first project. This is for analysis of sequences. It's the name of the project in Portuguese. So we start that in 1988. So when the FAPESP launched the Genome Project, we have a small group, myself, to work, and Darcy to work in bioinformatics. We are a little bit familiarized with the bioinformatics works. So, and last year in 2000, we have decided together with Marie to celebrate the 20 years for the genomics. Why 20? Because the that the Shigella fastidiosa paper was published, and at the same time, as I will show, is the inspiration for the creation of another networks in Brazil. So we would like to celebrate the two events. And we invite a lot of, we intend to celebrate this in the beginning of 2000, but due to the pandemic, we realized this in the Sociedade Brasileira de Genética at the end of 2020. And we have many of the researchers that are here, or talk today here. It was present in this seminar. In this occasion, we have the opportunity to work with the two persons that are involved in the NCT for the divulgação científica, and to perform a survey to identify, we have to understand what is the past, the present, and in the genomics and in the bioinformatics in Brazil. So we collect information, we perform our survey for all the scientists that work in the bioinformatics in genomics in Brazil at this time. We had, I will show some results, and we have three main questions that we want to address. What's the profile of the research currently work in the field, and collect and evaluate the details about the genomics and the bioinformatics from the point of the view of the professional, and to collect the data from the distribution of geographic for this area in Brazil. And we want to know also what's the perception about the present and the future, how it's confirmed from about the thoughts that the person from Brazil and now in other countries. And what is the milestones for the area in the wishes of the participant and the students for the near future. So we have these questions, and I will present this result a little bit later. But everything, and then we decide to write an article that is something that Fernando Reinach would like to know about what's the experience of the bioinformatics and genomics in Brazil. And we, in this article that is published in the Genetic Molecular Biology, we start, of course, with in 1990, the history of Brazil. We start with the Shigella Genomic Project in 1997. And we use this in 1999, the Human Cancer Project that we'll present tomorrow by Ana Maria and colleagues, and is so important for the creation of the Brazilian Genome Network. We replicate the same model that was implemented in the state in São Paulo. We replicate for all over the country. So the Brazilian genome, you use, the coordinator was the same, Dr. Andrew Simpson, and Ana Maria helped a lot with the assembly and discussing, and Jesus participate also in the lab, and using the same methodology, we spread the experience, the fantastic experience in São Paulo, for 25 labs all over the country. From the north or the south of the country, we replicate the same experience. So, Luiz Fernando, thank you very much for FAPESP to conducting this project, and thank you for the Ministry of Science, Technology, and CNPq for not create anything different, only to replicate that what is a great experience, and I will show that Brazil is in a good shape at the moment. After that, in 2002, we create, we don't have, as many of you talk, a lot of bioinformatics in Brazil. And so we decided to create a specialization course during 1 or 2 years. And then at this time, CAPES creates 2 postgraduate programs, one here in São Paulo, another one in UFMG, and so we start to the formation of human research in Brazil. After that, we create our society, AB3C, a post-graduation course around another states, in Rio, one at LNCC, other in Fiocruz, after in Rio Grande do Sul, and later on in Rio Grande do Norte, and Universidade Paraná, no Paraná, also in Paraná. And then, so that is the panorama of the bioinformatics and genomics in Brazil until 2016. And what about Latin America? How is Brazil when we compare with the 19 countries in Latin America? So you can see here in the first bar that Brazil is the country that the most published the most paper when we search for the words bioinformatics or computational biology. Brazil continues, as Pérez told us, the beginning of the genomics, carnival, etc. Brazil until 2019, this is a paper from the SoyBio network that Brazil belongs to that network. Brazil continues to be the country, one of the most important countries. And we can see here in this cloud, word cloud, that expressions, analysis, genes, computational comparative genomics is one of the words that's more present, and also the organisms that are most studied are genomes, most of them related to neglected tropical disease. That's important also for the regions. And when we see the connection of the countries, the collaboration between the countries, Brazil continues one of the countries that has more collaboration with another country in Latin America, mainly with Argentina, that is a partner for a long date, and Brazil continues to be a leadership in this area, and also this area, as in the rest of the world, is growing, as you can see in this bar plot B. Well, in this new paper that has been published 2 years ago in bioinformatics, we also comparing the country that's published, the country that publish absolutely or relatively when we compare the publication by million of inhabitants, that Brazil continues to be one of the leaders in publication of papers and cited papers all over the world. We can see here that the position in Brazil we continue to be in the top 30 countries with most publication all over the world. And you can see that in the Latin America, Brazil is the only country that continues to be part of this list. So Brazil is in a good shape, and we start, I reinforce, we start in 1997 with the vision of the FAPESP to start the genomics and bioinformatics in Brazil, and the whole community is working in this. And as we know, everybody knows here in this room, most of the methodology from in the growing of the genomics and the bioinformatics, the both science are working together and continue to growing together, is related not only with the equipment, the size and the quality and the amount of sequence can be generated for each different sequence, but also the quantity and the amount of the data that is generating by these sequences. And when we see the growing of the quantity and the amount of the data that is generating, mainly in the last thinking we will have in a few years, in 2025, I think this is in 2025, we will have something like zeta base pairs that we have to do, that we have to work from the sides of, from the bioinformatics. So, and yes, genomics, FAPESP has invested in a good field, because genomics now is greater than the Twitter, the amount of data generated by Twitter, astronomy, that was in the beginning, that was the science that with most amount of data that has been generated in the past. We pass the YouTube at the moment, and we think that we will have more or less, regarding to the genomics data, more or less 40 petabytes of data is being generated. And this is a quality of data that we have to collect, we have to process, that is spread all over the country. Oh my God. And then it's a good, also it's a good investment to do, to work in bioinformatics, because the bioinformatics area, when we go to the server, the global servers, is growing 11%, and is a service market that is estimated to the value in 6.44 billions in 2028. So FAPESP invests in a good thing, in genomics, in bioinformatics, and here is the future, as Paulo talked in the few moments. So I will skip that. This is the only slide that will show what we are doing, we have done in the past, what we are doing now at the lab. We continue to collaborate with many groups in Brazil. Within the Estado de São Paulo, we collaborate with different and many groups, and what we are doing is generate data in the lab for genomes, transcriptomes, small RNA, exome, metagenomics. We work a lot of metagenomics at the moment, and what in different areas, animal, human, health, biodiversity, biotechnology, and as we have a supercomputer, we can use the cloud, and we have the HPC for process and store this data. So I will not go in that. So I will pass from the last part of my talk. So is the results of the survey that we have performed, and I think this is important for the policy for the next year, for the FAPs and the government, CNPq, CAPS, etc. This is a new area. We start 22 years ago, 25 years ago. And we can see in the first A that the number of the participants when we have by gender is more or less is the same. So as we have the quantity of women and the males and females are the same. And in the part B is the number of participants or the students that or researchers. You can see that São Paulo is the state with more, we have more persons working by informatics and genomics related in Brazil. And in the age is very interesting because it's a new science. So we have people that working with different ages and with different expertise. So we have people that work for more than 10 years, and they have, most of them, they have the PhD. We have another part of the students or the researcher that is working it's a new area, so it's a lot of people that are interested in this area. And the perception for the participant, what they think about the area of bioinformatics and genomics, they thought that it's an area that is growing, that is promising, that is research. So it's a positive answer when we ask what's the area at this moment. And we can see here when we analyze the perception of the response for the survey, that most 46% they are thinking that they are extremely promising, and for 40% that neutral, and only 13% that's negative. When we see the milestones from the genomics and the bioinformatics in Brazil, I think the milestone, that's very important that we divide in some categories. In one of them, 87% of the responses were about the implementation, the programs, and the projects in the area. So the project that was implemented in the beginning by the FAPESP, we should renew this project. We should lance another project to collect more people, because the students, the perception that we need more programs to work in the network way. And also the technology, new technology, mainly high performer computer, that is a bottleneck for the area. I think from the sequencer we have at Brazil, we have a lot. And of course, the funding to support these projects. When we see the wish list, we most of the sponsors need a funding from the research and development in the area. And it includes the reduction of the price, and we have in the last few years, we had a drop of money from science and technology, so it's more or less obvious that is a wish list for the sponsor. But what is very important for me, from my point of view, is the formal education. Most of the sponsors ask for more courses, not only bachelor, but also PhD, that they want to conduct. So this is to show that FAPESP is the best foundation FAP in the Brazil. So it's responsible for most of the fellows and from in Brazil, not on the agency, for bolsters from the agency, more than the other agency, but also to support projects. So I think it's a challenge for FAPESP it's the next step to lance the new paradigm, the new challenges in the area in bioinformatics and genomics. As I can send you the paper that we wrote, that's a need from the community, for the researcher, and mainly for the students. Most of the students are going abroad. We have a fuga, the famous fuga de cérebro. At the last 2 months, I had 2 postdocs that left to another country. So we are teaching, we are forming, we are investing in many projects. We have to keep these students with a good quality of science that I show that we have done. So I'm here to celebrate the Genome 20+2 and to take it to FAPESP. Thank you to FAPESP to start this process in Brazil, and I'm able to continue to collaborate with FAPESP. It's a pleasure to be here. It also is an honor to be here, also representing a little bit outside the state of São Paulo. Thank you very much.
Thank you, Ana. Let's move on to the next speaker, João Carlos Setúbal. João was one of the 3 J's of the bioinformatics groups of the ONSA. João Setúbal, João Meidanes, and João Paulo Kitajima, who joined later the group. João, after the genome project was over, he joined the Virginia Bioinformatics Institute and the Department of Computer Science of Virginia Tech from 2004 to 2011. And fortunately, he came back, and he's now a full professor at the Chemistry Institute of the University of São Paulo. Please, João.
Boa tarde, é um prazer estar aqui. Good afternoon, it's a pleasure to be here. Thank you for the invitation to present this talk and for organizing this conference, Ana Maria and Marianne, or Anne-Marie and Mariana. It's a coincidence of the names. So I will talk about work that I have been doing for the past 8 years or so, microbiomes at the São Paulo Zoo. And since the Zalala project ended, the main new topic, at least for me, has been what we can call metagenomics and microbiomics. And this is the overall subject of this talk. But before I describe what I have been doing, I would like to present a few definitions. Microbiota is the microbial community in a given environment. The metagenome is the result of a metagenomic sequencing, no isolation of a sample. And the microbiome is the ensemble of genomes, gene sequences, and gene product sequences identifiable in a metagenome. These definitions, they are a little more strict than some other people adopt. But for the purposes of this talk, these are the definitions that I will use. So it's also important to state the difference between two kinds of exploration, sequencing explorations that you can do with microbiota, with a microbial community. One is the targeted or amplicon sequencing, in which you choose a specific gene to sequence. Usually you choose 16S for prokaryotes or ITS for eukaryotes. And the problem with that is that species identification is difficult or impossible. But it's still a tool that's used for many people around the world, primarily because it's cheaper than the alternative. And I've seen the term meta-taxonomics described for this kind of approach, but I'm not sure that it's common. The one that underlies this talk is the total DNA or shotgun approach. This allows you actual access to the microbiome, and species and even strain identification are possible. And in what follows, one of the main things that main topics that I will explain is the metagenome assembled genome. So metagenome assembled genome is a genome that you can recover from a metagenome. And Paulo Arruda briefly mentioned this in his talk, the one student who recovered 500 genomes. I assume that they're all MAGs from those samples. And I don't have time to go through the way you assemble MAGs, but basically you sequence your samples, and then you check for some profiles of small chunks of DNA called k-mers, and you also check the abundance, and then you put all these together, and then you do the binning, and out of the binning comes the recovered genomes from your samples. And it's also important to mention that MAGs, when I started working with MAGs, I was a bit skeptical of their value, because they could be, I could see that maybe they were artifacts. It could be very easy to get artifacts from the whole process. But now I have the view depicted in this slide, that MAGs are simply at the left end of a contamination spectrum. At the right end, you have the single-cell genome, which is the purest genome that you can get. And then in the middle, you have the traditional isolate genomes, so the Xylella fastidiosa that we sequenced 22 years ago is an example right in the middle, and the MAGs are on the left. Okay, there has been an explosion in MAG sequencing, and I will just show you 3 articles that have been published. So there was this first one back, I don't remember the date, something like 2017 or so, 8,000 MAGs, and then this year or last year there was this catalog of over 5,000 MAGs from the gut microbiota of animals, and then very recently 32,000 metagenome assembled genomes from infant gut microbiome. So this gives you a sense of how much emphasis is being put on MAGs. So the subject of my talk is this MetaZoo project. It's a thematic project that was funded by FAPESP between 2013 and 2018. It was a collaboration between the University of São Paulo, my host institution, the São Paulo Zoo Foundation, and Unifesp. And the idea to do this came from Luiz Juliano Neto, who used to be a professor at Unifesp. And the way he tells the story is that in 2009, he hosted a visiting professor from Japan called Kohei Oda from the University of Kyoto. And he took Oda to visit the zoo as a tourist destination. It was not part of the scientific visit. And for some reason, they could get to see the compost operation at the zoo. And Oda told Juliano, well, this is a gold mine. And Juliano immediately noticed that this was an important observation. And so he took that and moved forward. And he proposed the creation of a microbiology lab at the zoo. And he got a FAPESP grant to studying the compost at the zoo. And that was basically a pilot. And the thematic project that I had was a successor to this pilot. So this is the composting unit. It's a semi-industrial operation at the zoo. And it was part of the thematic project also to study 2 other environments at the zoo. One was what we usually say the zoo lake, but it's actually, the official name is the São Francisco Reservoir. And also there are many howler monkeys in the zoo, both free living in the forest that surrounds the zoo, as well as captive individuals. And it was also part of this project to sample feces from howler monkeys and compare the microbiome between free-living individuals and captive individuals. So most of all, I essentially will say everything I'll say is about the compost. The pilot results were published in this paper here in 2013. And the main lesson that we got from that pilot was that bacteria dominate compost. For those of you who are not familiar with compost, it's a process that is driven by a microbial community. That's why it happens. And so generally, people already knew that you could have bacteria as well as fungi. But in the case of the zoo, it's totally dominated by bacteria. And there's nothing mysterious about the fact that it's bacteria that dominate. It's just because the temperature that the composting reaches, which reaches above 70 degrees Celsius, it kills all the fungi and only the bacteria survive.
There was a follow-up. We published this in 2016. And this was a much more extensive. I said the other one was a pilot. This was a much more extensive, and this is given in the title. So you see there that there's metagenomics and metatranscriptomics, and also the word dynamics is very important because we were able to follow the composting process, which takes about 3 months. So we sampled over time. And our goal was to understand not only the specific microbial community that was acting, but also how that community changed over time. And we also did the study of a consortium because the actual compost in the zoo is a very complex problem, very complex environment. And so you have to do something in order to simplify it. And one of the things that you can do is extract a sample, take it to the lab, and cultivate in the lab in a consortium way, as opposed to individual bacteria, individual species isolation. So we did this, and we also published this paper in 2017. And finally, last year, we published another follow-up study in which our focus were the MAGs. And this was published in BMC Genomics. And so I will now briefly describe the results of this last paper. We had two series of samples. One was called ZC4. And for that ZC4, we obtained 49 high-quality MAGs. You have the 3 different colors there. The colors means, the blue means those are MAGs for which we could identify as species. The orange ones are the MAGs for which we could not identify the species, but we knew that there were MAGs in other samples, in other projects around the world that were very similar to those MAGs. So I call them conserved hypothetical MAGs, in analogy with conserved hypothetical protein that we are all familiar with. And finally, on the left, you have the H-MAGs. These are the hypothetical MAGs, and they're presumed to be the completely new species. So you can see that almost half of them were completely new. So the dynamics, this is a figure from the paper that I mentioned. It synthesizes the knowledge that we got by following up the composting process through all those samples. I won't have time to describe the different aspects of this figure, but it suffices to say that the different circles there represent the MAGs. The different rectangles represent the days. So you can see we had samples from day 1, day 7, 15, 30, 78, 99, so almost more than 3 months. And in each rectangle, we depict what we thought was the main thing that described what was going on in composting at that point. And 1, 3, and 64 are together because at day 64, there was a turning of the compost. And we verified that with the turning, conditions became very similar to the beginning of compost. So this graph shows metatranscriptomic abundance for 4 MAGs. And the 4 MAGs that you see, and on the x-axis you have time. So here, time 0 on the left and time 99 on the right. And there are 4 MAGs there, and the reason I'm showing you this is because we noticed that these 4 MAGs, their abundance increased at the very end of composting. And that was important because at the very end of composting, basically all the nutrients that you have left is based on lignin. And lignin is the material that is most difficult to degrade. And so for the biotechnology industry, it is important to find good lignin degraders. And of those 4, those 2, number 8 and number 20, appear to be good lignin degraders based on the fact that we could look at the genome and then we could see that they have genes that code for lignolytic enzymes. MAG number 8 was already known, Pseudomonas thermotolerans, which means that someone else, somewhere else, had isolated this genome, but MAG 20 is a novel gamma proteobacterium. So presumably, this will, we will take this forward and find out more about this too. So this is still ongoing, meaning the work on the composting data is still ongoing. We have new samples from inocula and from the mature compost. We are now a little more liberal with the criteria for recovering MAGs. For the BMC genomics paper, we were very strict in requiring 80% completeness, but now we are allowing 50% completeness, and that's because everybody uses 50%. There was no point in us being very strict and keeping to 80%. And so the current MAG numbers, given that, those new samples, as well as the more liberal criteria, have resulted in a total of 166 MAGs. And just like Paulo, I have just one student taking care of this 166 genomes. Those 166 genomes are not all different, and this circle shows you the relationship between them. So whenever there's a line going from one point to another, that means that under certain similarity criteria, we can think that these two are the same. So in other words, we see the same genomes showing up in different samples. Now I'm nearing the end of my talk. For the other two projects, all I have time to do is to show you the number of MAGs that I got. So for the San Francisco Reservoir, we had 51 high-quality MAGs, and you can see that the vast majority are new, which was a kind of a surprise. And for the howler monkeys, also more than 3/4 are new MAGs, which means that we have a lot of novelty there. So basically, this is the end. I would like to acknowledge Luiz Juliano Neto, who, as I said, was the person who had the idea to study the compost. Also, João Batista da Cruz and Paulo Bressan at the zoo, João Batista was the scientific director of the zoo at the time, and Paulo Bressan was, and I think he still is, the president of the foundation, and they were essential for the collaboration to happen. And there were a lot of people who participated in or still participate in the MetaZoo project, but I would like to emphasize these 6 people here. So Aline Maria da Silva is my colleague at the University of São Paulo, and she was a co-PI in the thematic project. Laila Martins is a researcher at the University of São Paulo, and Júlio Oliveira is a professor at Unifesp. All 3 are still active in the ongoing project. And the 3 below are either students or former students. So Davis Barbosa was my PhD student. He focused on the reservoir. Raquel Franco on the howler monkeys. She's defending her PhD thesis in a few days. And Susana Guime is my current PhD student, who's the one who takes care of the 166 MAGs that we are still studying. So funding and support was FAPESP, Zoológico, CAPES, and PQ. And with that, I thank you for your attention. I won't be happy to take any questions because I can't. Yeah.
Thank you, João. Let's move ahead. It's a pleasure to introduce you to our next speaker, Claudia Dorello. Claudia was, at that time, the ONSA's project, she was a young researcher, FAPESP's young researcher, jovem pesquisadora, within the ONSA initiative. And she played a very active role in the annotation and assembling as well, helping to assemble the genome of Xylella. But not only this, she also was very active in the sequencing of various other bacterial genomes and plant EST projects within the Agronomical and Environmental Genomes Initiative. Well, after that, she joined the Swiss Institute of Bioinformatics as the coordinator of the Swiss Brazil project. And, well, Claudia was a young researcher then, and now she's a young associate professor at the Department of Genetics of the School of Agriculture at the University of São Paulo. Please, Claudia.
Thank you very much, Mariane and Maria, for inviting me here today. Thank you, Aranha, for the nice words about me. I was surprised because in my crachá is written jovem, so not as jovem anymore. But I think I like to say, first of all, it was a great experience to be part of the Xylella project. I think I never had been anything like it after that. It was really important, and I thank FAPESP and all the colleagues here that helped me actually understand what I want to do for my career in science. And I became a genomics fan, and I've been doing this since then. And today, I will be talking to you about the work that I am doing in Zago, at Zago, in my lab. And there's a lot of, I'm going to explain a few things, but there is a lot more that we've been doing there. But this is just to show you how happy and involved I was after the Xylella project. I think for those of us that worked within that group at that time was great, really great opportunity. I felt that I was in the right place at the right time, and this is not something that happens very often in a person's life, so I am really thankful. Well, I'm going to talk about multiomics to identify components of the sugarcane smut molecular crosstalk. This is a research that I've been working on at Zago since I was hired there quite a few years ago. And let's see if I can. Oh. Well, I'm going to talk then without it anyways. Well, smut, it's a disease. Oh. It's a disease of sugarcane. Well, since the Xylella project, I worked with Professor Aranha in the genome of Leifsonia, which also a pathogen of sugarcane, and I kind of got in love with sugarcane. And with this, it's very complex genome, as I am going to show you in a few minutes. And this is the disease that I decided to work with. It's a very, well, I like to say nice disease, but the producers, they don't like me when I say things like that. But it's a disease that changes very dramatically the architecture of sugarcane plants. So we, from the top of from the apex of the plant, instead of having the development of the flower, of the flag in sugarcane, we have the development of this very long structure that looks like a whip. We call that structure whip, which is the first sign for the disease. So the fungus changes dramatically all the transcription of the genes in sugarcane, and instead the plant to produce the flower it uses the sugarcane machinery to produce this structure, which is part of the fungus reproduction. It's not the sugarcane reproduction, but it's now the fungus reproduction, and it's composed by a mixture of tissues, of cells from sugarcane and also from the fungus. So it's really, really interesting to see how a very robust plant, sugarcane can reach like 2 to 4 meters in height, being reprogrammed for the fungus to reproduce itself. So it's from the biology point of view, it's a really interesting system to study. Smut is a disease that is everywhere. It's the most important disease for sugarcane nowadays. There is no resistance. The fungus, even though sometimes the plants cannot develop the whip, and nobody knows why, the fungus decides not to sporulate in a plant that we call resistant. The fungus is in there. It's growing inside the plant. We just don't know why the fungus doesn't start sporulating to produce that structure. This is one thing that I am trying to understand also, one of the things that I'm trying to understand. So as I mentioned, it's everywhere in the world, and people from India and China and Australia are my main competitors because I keep receiving manuscripts to review from them, and I am always running behind them because my group is very small, and I think they have a lot of more money and people that I do. But as you can see, it's everywhere where we have a sugarcane field, even in Papua New Guinea, which is the center of origin of sugarcane. And to study this disease, we have to face two very important problems. One is the genome of sugarcane, which is really crazy. It's a genome that has, well, first, this sugarcane in the modern varieties that we have, it's not one species, it's a hybrid. The modern species are a hybrid, mostly of two species, Spontaneum and Officinarium. Officinarium is the species that gives the sugar, the noble cane that it's called, and Spontaneum is the one that gives resistance and robustness to the sugar cane. And nowadays we know that the genome of sugarcane is composed more or less the modern varieties. They have close to 90% of the genome is Officinarium, and 10% is Spontaneum, and we have some recombinant chromosomes between the two species. So it's not a very easy genome to study and select candidates to understand resistance, because most of the time resistance is not a one gene feature. It's not the result of only one gene. Only few cases we have major R genes that we call major resistance genes producing resistance to a given crop. And especially in the case of smut, this is not the case for sure. And the other problem is The fungus itself, it's a biotrophic fungus, although it's not as bad as the fungus that are the cause of rust, because we have a part of the cycle of this fungus. We can see it in the lab. I can have some work with the fungus in the lab, but the teleospores, the spores that are the responsible for dissemination of the disease, we cannot obtain in the lab. It needs to be in the plant, okay? We need to inoculate plants and have the plants to produce, or the interaction producing spores for this fungus. It's a fungus that penetrates. The fungus has lots of different features as when we compare with any other pathogenic, plant pathogenic fungus. It penetrates via meristem. As you can you imagine, it's important for the fungus be in the meristem because, as I mentioned, the symptom of the disease, it develops from the meristematic region, right? Instead of having the flower, these meristematic regions produce the whip, okay, where the fungus is producing its spores. So we did a lot of study first. Well, I decided to work with this, but like 10 years ago, we didn't have a lot of things about this fungus, so I had to revisit all the old papers and almost try to do everything again that was done before, but with new techniques such as using GFP market transformed fungus to see the fungus inside the plant, and using methods for looking at the fungus in electronic microscopy to see what's happening in there, in that meristematic region, what was happening there. So afterwards, when I see the genes, when I look at the genome, I could try to relate to what I was seeing in the plant, right? So we did a lot of microscopy, and as you can see, the fungus can grow in between the cells. It can grow growing inside the cell, but not disturbing the membrane of the cell. It doesn't produce a real austorium, which is the structure of the fungus that's responsible for the changes with molecular changes between fungus and the plant, right? The austorium, the fungus uses the austorium to recover nutrients or anything that it's needed for its survival. And also, the fungus injects molecules inside the cell so to disguise itself or to avoid the immune system of the plant. So I am really interested in understanding the stock between these molecules to do my best to decide which candidates, which genes are important for this interaction, so I can do experiments in sugarcane, since, as you saw the genome, it's not very nice to work with. And one more thing, usually a good pathosystem, the disease needs to be detected few days after inoculation. And in this case, we need to wait like 30 to 40 days, sometimes more, to see the development of the symptom. So you see, I have huge challenges to work with this pathosystem. Well, since I started work, I then decided, well, I need to understand the fungus and I need to understand the sugarcane genome. I was part of several initiatives to work with the genomics of sugarcane, so I knew a little bit of the genome of sugarcane, but I didn't know anything about the genome of the fungus, and I didn't know about the biology of the fungus, so the first works that we did was about knowing the fungus. First, diversity around the world, around Brazil. So I tried to get isolates from everywhere in Brazil, in different states, when we have sugarcane being cultivated. And we were able to define 2 different haplotypes, and these 2 haplotypes, they also have differences in terms of virulence. You know, they both produce, well, that was too fast. Well, they both produce a lot of, they both produce the whip. However, one of them is much more virulent, producing lots of whips. And then after that, we sequenced like 5 or 6 fungal genomes, completely sequenced these genomes from telomere to telomere using technologies, mixing technologies, PacBio. We mixed PacBio, Illumina, and more recently we are using Oxford Nanopore, the mini-ion, and works really nice for this fungus. It's not a very large genome, so it's very nice to sequence. And we did a lot of, with the candidates that we selected from combining genomics and transcriptomics, as you can see the other side, combining transcriptomics. We did some experiments now in the lab trying to prove that what we were seeing were related or were associated with the disease. But what was going to talk about today, I don't know if I will because the time went too fast. It's about the genome survey of resistant genes in sugarcane and the construction of a network, of a biological network based on Arabidopsis to understand the network of events happening during the interaction, okay? So I'm going to talk very quickly about it. And as we all know, we have that the first line of perception of a pathogen, it's in the membrane of the plant cells where we have the RGA proteins, the resistant analog proteins, and that are responsible for perception of proteins or molecules of the pathogen. Then we have a very nice signaling cascade, right? That reaches the transcription factors, and the transcription factors, they will start the transcription of genes related to resistance or susceptibility, right? So my idea, or our idea in my group, was to define the whole set of RGAs, the whole set of MAPKs, the whole set of transcriptional factors, and the whole set of PR proteins using the sequences available for sugarcane. We did that not only for sugarcane, but also for the sorghum bicolor. We did that for the R570, which is the monoploid version sequenced for sugarcane. It's a monoploid version sequenced for sugarcane, for sorghum bicolor, and for the spontaneum genome sequence, Saccharum spontaneum. Remember I told you that 10% of the genome of the modern varieties are spontaneum. And we also did that for a collection of transcriptors that we built based on Cardoso Silva in our first transcriptomic data. So we have all this genome, and we try to organize the sequence based on orthology. We used OrthoMCL to group orthologs and make sure that we have the same type of RGAs in each of the groups, okay? And after that, we place them in chromosomes, in the chromosomes versions that we had for each genome. And what we found out is that we have in chromosome 5, In chromosome 5, I can't point, but in chromosome 5, we have most of the RGAs clustered, responsive to SMUT, okay? This was the first thing that we saw with this data. But I didn't want only know about the RGAs. Né? I wanted to know the events leading, after the percepted, leading to the expression of PR proteins. So we started using and approached not co-expression, as everybody does, because I didn't have enough time points for doing a co-expression network, but we built a metabolic network based on Arabidopsis, using KEGG and all the information of the database BioGrid that contains data about protein-protein interactions, okay? So, and also we did one more thing that was search for terms in SwissProt. We did a text mining, and we also added to our network the terms related to defense mechanisms. The time is over. I'm just gonna show you very quickly. The results are really nice to see. If you can see, this is our network. Of course, we have like not the whole genome of sugarcane, which is paired with Arabidopsis thaliana, but we were able to find 9,000 more or less proteins. And here we selected a subset of modules that you can see that in the susceptible genome, all the genes are downregulated, and in the resistant variety, all the genes are upregulated. Okay, so and the question that I did for my database was, for my network was, I want to know a module that is very contrastant between the two genotypes, between the two transcriptomic data, and that have RGAs in it. So with that, I've been able to select candidate genes, very important for perception of the pathogen, and as well, for somebody that works with plant pathogens, we can see several gene names that are really important for perception of pathogens. And we did that not only for sugarcane, but also for energy cane. And we have the same kind of results, which means that we are hitting something very nice in terms of response to SMUT using this biological network. And, well, we are doing networks for the fungus also. We have a few candidates now. And what we are doing with these candidates is trying to, well, we want to improve our network with more and more layers, so we can provide genes, candidate genes for transgenic plants. This is what we've been doing now with IAC. The people from ISC's Centro de Cana, they have some of our genes to do transgenic and edit the genome also. These are the people working with me. It's a small lab, but I have lots of students. They are really good, very talented young people. And they are the ones, I just give ideas, and they are the ones that really do the work. And I like to thanks all my Xylella scientific partners for life, I think, I hope. Silvana Kresch, Luiz Eduardo Aranha Camargo, Mariane Vandes Luiz, João Paulo Kitajima, that still helps me a lot when I am in need for bioinformatics advisor, which is always. And I'm also working now with Grambio and with the person of José Bresciani. I think Grambio now was incorporated by NUCID, I think, but they are still interested in work with us, which is really nice. So That's it. I'm sorry that I passed the time. Thank you.
Thank you, Claudia. Now it's a coffee break now. We'll have to come back by 4:20. Okay, be sharp on that, please. And before you leave, we ask you to take your belongings with you. Don't leave anything in the auditorium because we're going to lock it. Okay, please? Yes, take it with you. Yeah, take everything with you because we're going to lock the auditorium, okay? And we'll be back at 4:20. Don't forget to visit the exposition while you eat pão de queijo. I'll see you in a while. Okay, let's resume our session. This is the last presentation this afternoon. We will watch a recorded presentation by Mark Blackster. He's a biologist and professor of evolutionary genomics at the University of Edinburgh. Since 2019, he's a leader of the Tree of Life program at the Sanger Institute. He works in the genomics of parasitic nematodes, but also of mammal viruses, and on topics such as the dynamics of infection.
Good afternoon. This is Mark Blackster from the Wellcome Sanger Institute in Cambridge. I'm sorry I can't be with you today. It would be wonderful to be there to celebrate 20 years of the genome sequencing program. And those 20 years have actually encompassed a lot of my genomic sequencing life. And I remember fondly collaborating with many Brazilian scientists in the WHO Parasite Genomes Project when they were involved in sequencing Schistosoma and Trypanosoma cruzi and assisting all of us in analyzing and understanding the genomes of those parasites. In those 20 years, genome sequencing has come along a huge way, and I now have the privilege and honor to lead a major project at the Wellcome Sanger Institute, which is trying to sequence the genomes of all life. And that's what I want to tell you about today. So I'm going to present my slides, and then we transfer questions afterwards. Okay, so what I'm going to do is give you an overview of the project that we're running, and a little bit about our methods, and then a little vignette, a little story about the biology of some of the species that we've been genome sequencing. So we live in a time, as you know, of a biodiversity crisis, and this biodiversity crisis is partly anthropogenic in terms of our impacts on wild ecosystems and on agroecosystems, where increasing urbanization and disparities of food for land for food means that wildlife is at risk. And the other major challenge is, of course, climate change. And if we fail to preserve the biodiversity that sustains this planet, we fail to preserve ourselves. Not only that biodiversity, but also human society relies on the ecosystem services of all the species that we share the planet with. And so we hope that genome sequencing can serve to help mitigate this crisis, can help to find new ways to stop and halt biodiversity decline, and also possibly supply new data that will fuel new bioindustries which are less destructive of the planet. As an evolutionary biologist, I also really think that these data are going to be absolutely intellectually, scientifically fascinating, revealing the tree or the trees of life, revealing how genomes and genes evolve, and understanding at a much deeper level the process of evolution. We stand on the shoulders of a major mega project in biology, the Linnaean project. This has been running for 250 years or more. It's involved hundreds of thousands of scientists. It's involved the equivalent in today's costs of many, many, many billions of dollars, pounds, euros. And it's generated a universal language which we can talk about species. And this is amazing. So this means anywhere you are, you can talk scientifically about a species, and we can discuss what a species is, if you like, but we can discuss scientifically about the concept of a species, and we all know what we're talking about. So this LAME project is a huge success, but it is only the catalogue of the library of life. It tells you what books should be in our library. And so, for example, here on this picture from Linnaeus's Regnum Animali is, I've highlighted the badger, the European badger, Meles meles, which was in the first catalogue. And this tells us that Meles meles exists and allows us to link to a large body of work which uses Meles meles as the keywords, if you like, to do the search. But this is just a catalogue entry. And what we want to do is make it so that you can go to the catalogue of the Library of Life, find the species you're interested in, and then download the book. Download the entire text of the book of Meles meles. And I use Meles meles the badger as an example because, in fact, we have sequenced it. The genome is available for all in the public databases, INSDC, ENA, GenBank, DDBJ. It's a very good genome. It's chromosomally complete. And we have assembled both haplotypes. In this case, we sequenced a trio. We sequenced the offspring of two known parents. And so this genome is now available for anybody working on carnivores, on badgers in particular, how they've evolved, what their genome encodes. For badgers, we're interested in their immune system because in Europe they transmit bovine tuberculosis. But also badgers are very social animals. And so this genome is also immediately available for tracking and understanding population evolution in badgers. And it's openly available. We've published a short genome note that describes the genomes, and we've made the data public on public databases. So this is putting the text into the Library of Life. So if you like, not printing out the books, because that would destroy the forest if you had to print them all out on paper, but generating the text so that you can download the books from the Library of Life. So this idea is not mine, it's not ours, it's part of a collective idea, which is the Earth Biogenome Project. So this is a project which is hoping to revolutionize our understanding of the natural world, sequencing life for the future of life, sequencing all 1.8 million eukaryotic species on the planet. 1.8 million is a lot, and the badger is but one, but we, the Science Institute, felt it's important to start, and so we have started. So we initiated what we call the Tree of Life program. This is, if you like, a department inside the Science Institute, which Science Institute is all genomics, and we are biodiversity genomics within the generally human-oriented and pathogen-oriented work that the Science Institute does. So we're going to sequence everything. Well, not quite everything, and it's important when you do these projects, obviously, to have some focus. So we have several foci in our initial years. We're only 3 years old, to try and build the skills and the technology and the attitudes and the processes necessary to deliver biodiversity genomics at scale. Our flagship project, our main project, we call the Darwin Tree of Life project. This is to sequence all 70,000 or so eukaryotes that have been named that live on these islands off the west coast of Europe that we call Britain and Ireland. So these are terrestrial, freshwater, and marine species which are on the British and Irish species lists. And this is a collaboration, I'll come back to it later. This is a collaboration across the many institutions that share these islands with these species. We also have a project called the Aquatic Symbiosis Genomics Project. This is a global project with partners in many, many countries across the globe, where we're looking at the interaction between a eukaryotic macrobiont and its microbial symbionts, whether they're algae, like Symbiodinium and corals, or bacteria, like many of the deep-sea clams that symbiose with methane-fixing bacteria. We're part of the Vertebrate Genomes Project, which aims to sequence, first of all, one for every order, and then one for every family, and then one for every genus, and then finally one for every species of vertebrate on the planet. We're part of the European Reference Genome Atlas, which is a Darwin Tree of Life-like project, which is going to sequence as many as possible of the species that are on the European continent. And we collaborate with other projects, such as the African Biogenome Project. And we're all under this umbrella of the Earth Biogenome Project. So these are projects which are going forward, all of which have the goal to sequence reference quality genomes, genomes of highest quality possible of eukaryotes on this planet, and share them openly. We, of course, started just before the pandemic hit, and I know it hit Brazil particularly badly. In the pandemic, we were running at much reduced capacity, but it did allow us to build what we call the genome engine here at Sanger. So this genome engine takes an identified specimen from the wild, generates a DNA barcode, partly to help the identification, but also to help track that species through our systems, extracts DNA, and that's not a trivial thing, from all these different kinds of species with all different kinds of tissues and contaminations that can happen. To generate long-read DNA sequence, we use the PacBio Sequel2e platform for the majority of our work because we found that it assembles best. We also use ONT when needed. We generate long-range information. So this is mostly high C. We use the ARIMA kits for high C, and this is to scaffold the contigs generated by the long reads. And we also generate a small amount of transcriptome data. So this is not to do functional genomics, but this is to provide information for gene finding. So those data are QC'd, and we make all our data QC public as we generate it, and then we generate assemblies. So while the sequencing is the revolution that makes us able to dream of doing this project, the other revolution that's happening is in assembly algorithms. And we now have some amazing assembly algorithms, and there's very good people to run them, which can assemble data at scale. So we first do a primary assembly of the long reads to generate contigs, aiming for a contig N50 greater than a megabase. And usually we're achieving better than that. We then scaffold with high C such that our scaffold N50 is greater than 10 megabases, where that's possible. If the genome is very small, we won't achieve that. And then we do curation. So curation is part manual, part automated, but the idea of curation is to turn the scaffolds into validated chromosomes. So associating chromosome arms with each other across highly repetitive centromeres, attaching telomeres to chromosomes, and sorting out any misjoins and misscaffoldings that the software may have made. One of the things we realized early on in the project is most of our tools are designed to work really well on humans or bacteria. And the diversity of genome structures that we're finding now is challenging those tools, and we're finding ways to improve them and make them work for all species. We then submit the data to ENA, where it becomes immediately publicly available. We generate the annotation in collaboration with Ensemble. So Ensemble find the genome, well, we tell them about the genome in ENA. They upload it to the Ensemble system. They perform repeat finding and gene finding using the RNA-seq data, and then publish openly the results of that, so the gene models and the repeat structures on the Ensemble Rapid website. All the data are made available through a Rapid Open portal, which offers links to all the data files, etc., so you can find them, and there's API access if you want to do it in bulk. And then we publish something we call a genome note, which is a short paper that says, this genome is sequenced, please use it. And when you use it, credit these people. And the people we credit on the genome note are the person who collected it, the naturalist who identified it, or taxonomist who identified it, the people in the museum who do our curation and archiving, the people who did the DNA extraction in the wet lab, the assemblers, the curators, and the annotators. So the idea is that this paper stands as an immediate citable object, so that it's clear, A, that the data are available, and they are, and B, who to credit for those data. And these small papers, they're very short, are, if you like, part of our open science. So this engine we built, and it's running at speed now. We're submitting over 50 species a week to the extraction process. And this is what happens. This happens to be an easily assemblable genome. It's only half a gigabase, so only 500 megabases. But this is what comes out of our automated assembly pipeline. We generate 25-fold coverage in high-phi SQL data. We generate about 50-fold coverage in high-C data. And our automated assembly pipeline generates an assembly. And the assembly has an N50, well above a megabase, essentially just subchromosomal. And the scaffolding turns this into a set of chromosomes. And if you count there are 31 squares of high-C interaction on this map. And those are the 31 chromosomes of this moth, the Lilac Beauty. And in the final assembly here, there's only one gap. So that's the sort of quality of assembly we can get in an automated way. There are still problems. Hymenoptera have wonderful centromeres. They're very difficult to assemble, and some genomes are much bigger and take a bit more engineering to get through. But in general, genomes are really easily assemblable. So we've challenged ourselves to say how high and how low can we go. So high means how big a genome can we assemble with this technology. The average size of genomes in Britain and Ireland, if you like, on these islands, is about 3.5 gigabases. I mean, it's so high, even though most of the species have smaller genomes, because there are some genomes there with very, very large genomes. And the largest in Britain and Ireland is the mistletoe. It's a parasitic plant which parasitizes many, many trees and is traditionally used at Christmas as part of a sort of pseudo-pagan ritual where you're meant to kiss your loved ones under the mistletoe, and this gives you lasting luck for the year. So we decided to sequence mistletoe, even though its genome is 93 gigabases. That's 30 times the size of the human genome. So we went ahead and generated 25-fold coverage in HiFi, this the PacBio HiFi, and that's over 100 PacBio flow cells, and 50-fold Hi-C. So that's a lot of NovaSeq sequencing. And I wouldn't be showing you this if it hadn't worked. So it's currently in curation. Curation is taking slightly longer. I'll explain why in a second. But this is the primary scaffolding, primary Hi-C scaffolding of the contigs. So the contig N50 is 30 megabases. And the total length of the primary assembly is 102 megabases. That probably includes some haplotypic duplication we haven't managed to remove. And the simple scaffolding with the Hi-C data generates a scaffold N50 of 6.5 gigabases. So that's 2 human genome size, total human genomes as a scaffold size. And the longest scaffold is about 3 human genomes in length. And the 3 little pink bars on the Hi-C map indicate 3 human genomes. So this is resolved beautifully into 10 chromosomes, and maybe a B chromosome present also. And the chromosomes all have a very typical structure of strong interaction between chromosome arms, and there is also some interaction between the centromeres, the particular pattern that we know from other large genome plants such as wheat. So this is the assembly of Viscum album. It is chromosomally complete. It will be chromosomally curated by Christmas. And when you're standing under the mistletoe, or you see here people doing that in Europe, you will know that actually that genome is sequenced, even though it's the biggest genome in Britain and Ireland. So we're unscared of big genomes now. There are still challenges. There are polyploid genomes, which will be more difficult to resolve. And there are polyploid, especially polyploid genomes that are partly rediploidized and partly still we're combining where there may be issues with resolution. But we're actually not scared of big genomes. You just have to sequence a lot. The other issue is size. So most species on this planet are tiny. Most individual organisms are tiny. We like to sequence from an individual because that gives us the ability to just be sequencing 2 haplotypes. And unlike us, humans, where genetic diversity between us is actually quite low, and most wild species have very high genetic diversity. And this makes for really complex graphs if we sequence more than one individual, when we're sequencing 4 or 8 or hundreds of haplotypes at once. So we try to sequence from one individual. But in very small organisms where there's maybe only 1,000 or 2,000 cells, there isn't enough DNA in those organisms to do the normal library prep. So we've been working on ultra-low input library preps. This method here is called PIMS. It was devised by Chris Lawmer, who's now at the Natural History Museum. And it uses a carefully controlled long-range PCR to generate DNA that's long enough or reasonable enough to sequence with PacBio. We also isolate the messenger RNA and do RNA-seq. So from the one small specimen, we're able to get a PacBio sequencing library, which is good enough to cover most of the genome, and also RNA-seq. And these assemble really well. We don't get as long inserts as you would do in a normal library, so they're only about 9 kb. That's the limitations of long-range PCR, but they're good enough for assembly. And this is work from Erna King in my group using Chris's method, showing the N50 on the x-axis and the Busco completeness on the y-axis of a set of genomes, 26 genomes she has generated from individual nematodes. These are marine nematodes for which we have no prior information essentially at all, apart from sometimes an 18S sequence. And the N50 of the genomes goes up to 600 kilobases. Most of them are around 100 or 200 kilobases. And this N50 is the best we can achieve with the method, we think, because the method, because it involves PCR, has real problems with amplifying inverted repeats in the genome. So these are very good assemblies. The Busko scores are low, but that's traditional in nematodes. Busko scores are often low. But these are very good genomes. They're better than most genomes for nematodes that have been sequenced so far. So they're amongst the top 20 or 30 nematode genomes out of the 200 that have been sequenced. But what Ern has then been able to do is take a number of individuals from the same species and generate high C data. So the high C doesn't have to come from the same individual because we're just scaffolding the two haplotypes generated from one individual. And with this high C data, in this case from 22 individuals of Odontocalamus, she's been able to generate a chromosomal assembly of this free-living marine nematode, first free-living marine nematode that's ever been put into chromosomes. And so this is now achievable across biodiversity. So we can do the very largest genomes, we can do the very smallest genomes down to about 1,000 cells. We still struggle with single cells. How do we sequence protists, if not from culture? And there are still issues which we have to overcome, but we're overcoming slowly. The other thing we get for free with these genomes is we get their cobiome. So the cobiome is what we call the group of non-target organisms that happen to be on the specimen we catch. So the specimens we catch, we usually would not take a very ill or very contaminated looking specimen, but they will carry parasites, they will carry pathogens, they will carry symbionts, and those necessarily get sequenced at the same time. And so we've been sequencing or co-sequencing these symbionts with the target. And this moth, Phalaera bucephala, happened to carry 3 different Wolbachia genomes. Wolbachia is an alpha proteobacterium that's a reproductive manipulative parasite. And we were able to sequence and assemble those 3 alpha proteobacteria into circular genomes. Okay, so we call these cobionts. In this case, we know they're probably pathogenic symbionts, or reproductive manipulative symbionts. But in other cases, we don't know the relationship between the bacterial genome or the protist genome that we assemble and the host, so we call them cobionts. And Emily Vancaster, whose work this is, has been doing this across the insects that we've been assembling from the Darwin Tree of Life, and she now has 110 new Wolbachia genomes to complement the 90 or so that are already in public databases. So she's doubled the number of Wolbachia genomes available, and 77 of those genomes are circles, so they are complete genomes. And this graph shows the relationship between the Wolbachia supergroups, clades of Wolbachia A and B, and the insect hosts in which they were found, showing, as expected, this mixing. And there's lots of other detail about these genomes and about the relationship with their hosts in Emmeline's bioarchive paper. We like making our data public, and we make our data public as early as possible. So yes, we submit the raw data and the assemblies into the public database as soon as they are complete. But before then, we have a data portal where you can go and see what we're doing and where we're at. And if there's a species you're interested in, you can ask us to do it. You can see our raw QC data. So these are data coming off the machines. We do QC and we publish that openly on our TOLQC portal. All the annotation and gene finding is available on Ensemble, and you can interactively scan the genomes through our blog toolkit viewer. The genome notes are all openly published in Welcome Open Research, which is part of F1000. So they're small data notes and they're fully open access. And we coordinate and correlate all our work across the globe inside the EBP through the Genomes on a Tree or GOAT data system, which again is openly accessible. So you can see what we're doing, where we're at with individual species, and what our plans are. And so here's the Badger genome note showing the quality of the genome, and the high C map to show the integrity of the chromosomes. And please, if you're interested in carnivores, or interested in badgers, interested in social behavior, please use the genome. So our major project, as I said, is Darwin Tree of Life, and I just want to quickly explain how that works. Darwin Tree of Life is this project to sequence everything in and around these islands. And it's a collaboration between 10 institutions, 10 organizations, some of whom are traditional biodiversity organizations, the Naturalist Museum, the Botanical Gardens in London and in Edinburgh, and Marine Biological Association. So these are organizations which traditionally monitor or collect biodiversity, but are not traditionally genomic. We also work with University of Oxford's White Woods field site, which is an ecological observatory, and they with us are turning that into a genomic observatory, where not only do they have ecological information about the species in this ancient woodland, but also the genomes of all the representative species there. We work with additional sequencing centres. The Earlham Institute and the University of Edinburgh are also doing sequencing, the Earlham especially on protists. And then we work with analytical partners, again, University of Edinburgh, University of Cambridge, Earlham and EPI are analytical partners. And this co-biome of partners is actually very symbiotic. So we all bring to this project components that each one of us couldn't do alone. We're co-funded by the Wellcome Trust, but each of the institutions also brings significant local funding into the project. Our goal in the first phase is to sequence one representative of every family of organism found in and around these islands. That's about 4,000 species. There are about 9,500 families of organisms on the planet described. And so just by doing Britain and Ireland, we hope to do about 40% of all the families on the planet, so giving a representation across diversity. We're also doing species that are interesting, important, or iconic. So these are species which might be the subject of conservation or preservation efforts, species that might be important to agro-ecosystems or ecosystem engineers, and species which are being worked on by others where having a reference genome is very useful. So we have got quite a lot of sequencing going on, which is not one per family. We've got some places where we sequence more than one per family, but the goal is to get to about 4,200 species with approximately one per family. Currently, we've got nearly 4,000 species in the freezers at Sanger. That number will soon go well over 4,000 as the late summer collections come in. So we're well on the way to that phase one goal. And this is where we are with sequencing. So this tree is a taxonomic tree, and the leaves are families. Okay, so we've got 864 leaves on this tree. 771 of these are from the Darwin Tree of Life project. Others are from things like the Aquatic Symbiosis Genomes Project and the Vertebrate Genomes Project. And the histograms on the outside show what we're doing. So blue is things that are in progress. For most taxa, there's one species in progress. Or red is what we've completed. So we've completed about 450 species for GTOL and about 570 species overall. And these species are generally distributed around the tree. There's a certain bias towards Diptera and Lepidoptera, and this is because of interest in pollinators and interest in climate change impacted species. And I'll illustrate some of that later. But anyway, the project is going amazingly well, and we're at over 500 species submitted already. So one of the things we want to do at Sanger, and many of our partners want to do, apart from use these genomes for conservation, is to do new biology. And it's amazing what these new reference genomes do for new biology. I've sequenced genomes for about 20 years before joining the Sanger, and the genomes I sequenced then are nowhere near the quality of the genomes we're getting now. So we're seeing all sorts of new biology in these genomes because they are chromosomally complete. And I just want to illustrate this quickly with an example from Lepidoptera. So Lepidoptera are one of the most speciose orders on the planet. So there are 180,000 species of Lepidoptera, which means they're 10% of all described species. So if we understand Lepidoptera, we've understood 10% of the biota. That's every species is special in itself. So I'm not suggesting simply understanding the deep structure of Lepidoptera gives much insight. And one of the things we've been interested in is the evolution of chromosomes in Lepidoptera. So this is the human karyotype 23. Why 23? Why not 24 or 27? How do karyotypes change? How do karyotypes evolve? So the human chromosomes, most chromosomes of organisms are centromeric, and so they have two arms and a centromere, and the centromere is what is used to organize the chromosomes during meiosis and mitosis. But actually Lepidoptera are holocentric, so fully 10% of all the species on the planet, and the many other species are holocentric, which means they don't have defined centromeres. And so one would expect their chromosomal evolution to be very different. We know that chromosome arms tend to evolve as units through life. But when you've got chromosomes that are holocentric, there's no technical issue or biological issue about breaking a chromosome in half, because both halves should still be able to segregate, and no issue about fusing two chromosomes, because if you fuse two chromosomes, there are no competing centromeres that would possibly rip the chromosome in two. So how do Lepidopteran chromosomes evolve? So we know from karyotypic surveys, this is an amazing piece of work by De Vos et al., that there is a median or modal size of the Lepidopteran karyotype of 31. This is obviously biased by what's been sampled and everything, but what's amazing also is that there's huge variation. So the smallest number of chromosomes is 5 in hypothyris, and there's one of the blue butterflies, the Lycaenids, called Polyomytus atlanticus, which actually comes from Morocco, which has 223 chromosomes. So huge variation in chromosomes, numbers, but a modal number of 31. So what's going on? What do we see from the genomes? So we've been sequencing Lepidoptera. Turns out they're quite easy to sequence, which is a lovely thing, and they assemble quite nicely. And the genomes are relatively small, so between about 300 and 1.1 gigabases. So we've generated 188 Lepidopteran chromosomally assembled genomes, and Charlotte Wright, a PhD student in my group, is now analyzing those for how they've evolved. So the following slides are based on a preliminary data set of 89 species. The general story that these 89 species is emphasized and underlined and confirmed by the analysis of the full 212. And I'm just going to take you through what Charlotte has discovered. So first of all, yes, 31, so the graph on the right is the chromosome number. 31 is the normal number in most Lepidoptera, and most of the species we sequence have 31 chromosomes. Lepidoptera are WZ, so the female is heterogametic. And where we can reassemble the W chromosome, it's often quite difficult. But some groups do have very different chromosome numbers. So for example, in the Lycaenids, in Lysandra, this goes up to 89 chromosomes. The number of chromosomes doesn't correlate with genome size. So genome size is the graph in the middle. Genome size varies quite widely, even within families, and we can have families which have absolutely the same chromosome number and very different genome sizes, and similarly within groups that vary in chromosome number, the genome size is approximately the same. So what we found is that we can identify ancestral linkage groups by looking at the genes that are on each of the chromosomes. So we take the species, we identify usually Busco genes on the chromosomes, and we correlate the chromosomes by the Busco genes that they contain. And across the whole of the Lepidoptera, we can use this comparison. There's a comparison here between Bistembettularia, a moth, and Melitea. We can a butterfly, where we can do one-to-one homology, one-to-one orthology between chromosomes. So there's very little migration of genes between chromosomes, and so we can define what we call Merian elements after Anna Maria Spillin Merian, who was a lepidopterist and entomologist in the early 1700s. She wrote a lot about metamorphosis, so we celebrate her by calling these Merian elements. So homologous chromosomes can be identified. We can say this is Merian element 1 in all these different species, so we have orthology declarations, and even gene order is conserved. So this is moth and a butterfly, and the fact there are diagonals on that plot says that the genes are in the same order. So chromosomes are boring in Lepidoptera. We expected them to be exciting because of the lack of centromers. They're not boring, they're very interesting. And here's a boring butterfly, Vanessa, where the fact that the entire graph is gray means that there are no genes that conflict with this Merian element designation. But there are rearrangements and there are fusions. So that the commonest kind of rearrangement is a simple fusion between two chromosomes. Fission is much rarer. The commonest type of rearrangement is fusion between two chromosomes. And on this tree, we've now colored where we've seen rearrangements, and those are usually fusions. And you can see that usually the fusion is on a branch leading to a tip, and occasionally the 7 orange dots on the tree, we can detect an ancestral fusion which has been maintained in more than one species. The quilt plot or cushion plot on the graph shows the interaction between chromosomes. And one thing we take home from that, just to say, is that it's often a fusion with the Z chromosome, the sex chromosome, that is commonest. So small chromosomes are more likely to fuse than our large chromosomes, and the chromosome that is most involved, except for the Z chromosome, which is usually the biggest chromosome, and it's more likely to be involved in fusions than any other chromosome. So there's a size bias to which chromosomes fuse, and there's also a bias towards the Z. The plot here is absolutely fascinating. The fact there's so much light color means that the data matrix is quite sparse. So I don't want to make too much of this other than just that small chromosomes are more likely to be involved in fusions. But there's some really interesting patterns here. And these patterns we hope to explore with larger data sets. Some species, however, undergone extensive rearrangement. So most species are boringly 31 or 30 or 29. But some species have undergone extensive rearrangements. And this graph shows 3 independent sets of rearrangements where, for example, in the Lycaenids, Cyaniris has the ancestral structure of 31, but the 2 Lysandra species have undergone extensive fission. So they've generated much, much higher chromosome counts by slicing ancestral Merian units. This is a similar pattern in Leptidia, but in Leptidia we also see fusion between fragments of the Merian elements. And in Pieris brassicae and Pieris napi, the Pierids, we see a large amount of fusion and then fission and then fusion, such that they have many fewer chromosomes than 31, but those chromosomes are made up of fragments of the original Merian elements. So this suggests there's several processes going on here. There's a counter, an abacus, which counts how many chromosomes there are and keeps things at 31. But then things can change. And so there's something that glues chromosomes together and something that slices chromosomes. And in the ones where we see chromosomes being glued together significantly, for example, the Pierids, they've been chopped first. So there's an independent slicing mechanism. And this is amazing. So in the large white, for example, the largest chromosome is made up of 8 fragments of 8 independent Merian elements, but those fragments retain gene order collinearity with the original Merian elements they came from. So there's also something that prevents internal rearrangements in Lepidoptera, and those of you who work on Heliconius and related species will know that there are very important structural rearrangements in heliconines which are involved in posomatic wing coloring. And so those are very special events in butterfly evolution. So once we have orthologous chromosomes, we can compare them. And so on the left-hand side of the graph are 8 example Merian ones from 8 different species, showing that across that chromosome there are patterns of, for example, in this case, repeat GC content, where it's higher at the telomeres and lower in the centers in most species, but there's one in the middle, the teal-colored one, which is relatively flat. The other thing we can say is that small chromosomes tend to have much higher GC and much higher repeat content than large chromosomes. So in every species we look at, there is a negative correlation between GC and proportional chromosome size. And this is across all species. So that the graph with all the lines on it, I love because it's got 3,000 dots, and every one of those points, every one of the data points is a whole chromosome, telomere to telomere chromosome. So every species has approximately the same slope. There are some that disagree, but this is a strong pattern in lepidopteran chromosomes that the small chromosomes have lower gene density, higher repeat density, and higher GC than large chromosomes. This is not what we expect because the smaller chromosomes undergo more recombination. There's one recombination approximately per chromosome in the males, and therefore we would expect the small chromosomes to be more efficiently purged of repeats than the large ones. It also means that the genes on the large ones have experienced much less recombinational mutagenesis through time and have been in much closer cis linkage with regulatory elements through time because recombination is less dense on those bigger chromosomes. So this patterns not only the chromosomes, but must also pattern gene regulatory networks and must also pattern gene evolution. And this is the same across species. It doesn't matter which species we look at, the story is the same. So stasis and dynamic change in these lepidopteran chromosomes. The 31 Merian elements at the base of the ditrysia, which is the set of species analyzed in this 89, and they've been generally conserved in most species. Where there has been change, we find fusions much more common than fissions, and the fusion rate corresponds to the size of the Merian element. But there have been, we now know, 8 independent origins of extensive genome reorganization. And these independent origins have different phenotypes, and these phenotypes tell us that there's 3 different processes at least going on. One is an abacus, which counts. One is a pair of scissors, which chops, so that something fragments the genomes. And another one is this glue, which fuses chromosomes together. And we're now, or Charlotte is now looking at the genomic biology of these 3 processes. So I get to talk to you, unfortunately not in Sao Paulo, which is a great disappointment. I get to talk to you about this amazing project, which it's my honor to lead. But actually, it takes a village to generate these data. And so, for example, for the Darwin Tree of Life project, where most of those Lepidoptera came from, they are working tirelessly to collect samples, to identify them, to barcode them, and to deal with the data post hoc in ensemble. And those people are just amazingly enthusiastic about this project, and it couldn't happen without them. But they also have collected an army, almost hundreds of engaged naturalists in Britain and Ireland who are collecting for us. So the experts who know their group of species really well and help us identify things. In Tree of Life, we have core teams who do sample management, wet lab processing, assembly, curation, and generally keep us organized, and they're essential to this whole process. And then in the Sanger Institute, we have the core facilities, both, if you like, the legal bit of doing Nagoya and wildlife permits, but also the R&D bit of adapting these technologies to our needs. But most of all, thanks to the long read team at Scientific Operations who do the sequencing. And then some of the data I've shown you is from work from my faculty. Charlotte did the butterfly work, Erna, Luis, Pablo, and Manuela did the nematode work along with Chris Lomer at the NHM, and Claudia and Emmeline were behind the all Bachia work. So very happy now, I hope, to take questions, and thank you very much.
I'll send a virtual thanking to Mark Blackster. We won't have questions because he's not online. And with that, we end today's activities. Thank you all for coming. And we convene again tomorrow at 9 a.m., beginning at 9 a.m. for the Cancer Genomics session. Okay, thank you all. Thank you.